First, I would like to extend my appreciation to all the presenters that will come in this session and all the presenters that did well in the precedent sessions. But before starting, I would like to mention or to make few announcements. So the first one is for the submission of the presentation. So to answer smooth running of all the sessions, I would like to ask you gently to give your presentations to the people at least one day, if not some hours before, so we can check if there is any incompatibility between your file and system or any technical issue of the projection. So that's the first point. So second point, it's I would like to remind everyone presenting the map of the country to check it, please, that the map is complete, the map of Morocco. This detail is very, very important. So please take some seconds to check it, or at least give your presentation before, so the technical staff could check it too. Third point is the gala dinner. So for those attending the gala dinner, it will take place at the Beauvois of Al Masira. A bus will depart from your hotel at 19:30. But for the people that are driving, please ask the organizing committee so to tell you exactly the precise location. It's when coming from Laayoune to ASARI, it's the 10th positions. So just please go to the organizing committee and they will give you all the detail. So last point, I said many points, this is the last one. So for the posters, for the poster session, be near your poster so you can explain it and you can tell people about your work. And even if you can, even during the coffee break, stay please near your posters because people, they would like to know more about your work. So saying that, I would like to start the session with the first presenter, which is Dr. Al-Amir Bushra, and who will be discussing evaluating and improving reproductive performance in the Moroccan camel herds, challenges and strategy. Dr. Al-Amiri, please, the stage is for you.
Thank you, Professor Badawi. Dear guests, I'm very honored to see you again today and to present you my topic, evaluating and improving reproductive performance in Morocco, camel herds, challenges and strategies. Sorry. The outline of my presentation will be as follows. I will start with an introduction, then I will give you the challenges for this sector in Morocco. I will present the reproductive performance and advances in reproductive manipulations, and I will finish my presentation with a conclusion. Let me remind you today also that camels in Morocco are the single hump type known as dromedaries. The total camel population in Morocco is about 200,000 heads, with over 80% located in the southern region of the country. They are raised about for milk and for meat. Even yesterday, you heard that a lot of this, and a large number of these camel heads are located in Dakhla, but you have to know that normally the owners are here in Laayoune, and the heads are located in Dakhla because Dakhla is the part of the country with a high rainfall, and this is why there is a large movement of this animal toward the Dakhla. But normally the large number heads are located here in Laayoune, as you see in this table. The populations, like I said yesterday, are about 3, the Marmori one, the Gurzni, and the Khawari one. The Marmori is estimated as a milky one, and the Marmori, the Gurzni one is recognized by, about its resilience. Like you see here, there is, Piru pointed out that there is a similarity between the Gurzni, Khawari, and Marmori. And the herds is typically a mixture of females from these populations, with one or two males selected by the breeders. But like I said yesterday, the herds are dominated by Gurzni type because the bulls used in these herds are generally from Gurzni type. There is many challenges in this. I mentioned some yesterday, but I would like to remind you about these challenges facing the camel herding in this south part. The first challenges are environmental and natural resources challenges, summarized as below. The region is with arid climates, and the impacts of climate change, we really feel it every day when you are visiting the farms. There is a scarcity of vegetation and its dispersion. There is irregularities in the feeding supply, and the feeding is based on rangeland and supplementation. The supplementation mainly consists of bread, unfortunately, which is the most common component used by almost all breeders. Normally, like you see, that's the bread is the most components, and there is also a mixture of flour and water called locally المريس. Another type of challenges in these herds are the herds did not express their full potential, as 40% of milk, for example, when we are talking about milk, is used for calves. Recently, 2024, Boujnan have made a nice work about milk, camel milk in Morocco, and he highlighted that at least 30% of this milk goes to the calves. The reproductive management is still neglected because we have this intensive system and rearing management as the main dominant, and the frequent movement of the herds make it difficult to follow and to put a good and a real reproduction management. The vast space makes it challenging to optimize technical supervision and extension services on camel, mainly, for example, for health. We have, till now, we don't have any clear program for health monitoring. Unfortunately, also, we don't have today, we don't have specialized shepherds. We are, all the herds are based on Mauritanian people coming with their own knowledge, and we are losing all the shepherds here in Morocco. We don't have any Moroccan. When we visited, if you have the possibility to visit the farm, we will notice that the whole shepherds are from Mauritania. For the health issues, from yesterday, you learned that we are facing a lot of diseases. We have this low health monitoring due to the vast area. The disease significantly affects the camel production in terms of quality and quantity. The reproductive disorders are more frequent in these herds. the knowledge and the training and knowledge, we are suffering from a lack of knowledge about the real genetic and productive abilities of the animals, the lack of traceability and follow-up of the products, as well as animal performance and health monitoring, the lack of traceability and follow-up the products, as well as the animal performance and health monitoring. Now, let me go now to give some definitions to make it easy for you to follow my presentation. The first thing I would like to emphasize is the fertility indicators, the reproductive intervals, and the reproductive losses. About the fertility indicators, we have to get pregnancy rate per cycle and pregnancy rates at the end, which the first one is defined as pregnant females at specific day, divided by all mated females. The second one is pregnant females in June, for example, divided about all mated females. As another indicator in the among fertility indicators, there is a live birth rate. For reproductive intervals, we have the age at first mating and the parturition interval, inter-calving interval. And the third one is reproductive losses, early pregnancy loss, abortion and prenatal mortality, neonatal mortality, and culling rates. All these parameters, I already say that. I will really skip this slides because the information I already put it in my first. And I go directly to this table, which summarizes all these parameters. And from this table, you see already that these parameters are not well studied because all of them in all works done till now in Morocco, they are just collected from surveys made by students, made by sometimes technicians. But here in ASARI, we are really planning and we are doing monitoring now for some herds to collect well these parameters and to define them correctly. And these parameters are not really different from that expecting and seeing in other countries. I will now present you some reproductive performance and the factors that affecting these parameters. First, I will go to the effect of seasonality. All people here in this room working on the camels know that there is an effect of seasonality, and changes are made in terms of testis volume, accessory sex glands, sex behavior, libido, testosterone levels, and semen characteristics. Old and recent studies demonstrate that when we are, for example, determining the profile of progesterone, of testosterone, the main hormone involved in sexual behavior of the males, we noticed, for example, from this table that the concentration is increasing from October to March. And this have recently shown when determining the profile of hormones extracted from the pole gland by Vias, who showed also that there is this seasonal effects on the behavior and testis gland secretion in males. This effects of male, you will notice it, for example, when you are examining the roots period. For example, in India, it's late September to March. In Sahara, it's October to May. Middle East, it's late October to late April. And in Morocco, we are in the same range. We are announcing from these surveys, not really good monitoring of extensive system. We are in this range October to late April. During the non-breeding season, the male camels exhibited no sexual behavior or interest in approaching ostrich females for mating. And from a local knowledge, we know that males that are loose in the herd tend to enter roots earlier and remain in roots longer than those managed more restrictively. The effects of season, like you know, all the scientists here, you know that normally to make the ovaries working well, we should have like a fuel. Then the seasonality, good season of reproduction coincide normally with the high quality of roorage, high quality on the rangeland. And the seasonality is it have a good effect on follicular ovarian follicular growth and maturation in camel. We have the age, the body content, many sets of factors can influence this. We have the age, the body condition, and the reproduction stage that influence ovarian activity at the beginning of the breeding season. Here in Morocco, Sagheri from 1998, there's a nice work on that, working on farm animals. Other factors that can influence the reproduction in camels, we found the nutrition. All of us, we know that nutrition is like a fuel for an engine. I think the nutrition is a good plan for ovaries and for testes to work well. Then for all organs for reproduction. The inclusion, for example, I give this sample example to remind you that these nutritional plans could have a good impacts on reproduction parameters. Mustafa, 2016, he showed that the inclusion of Atriplex shrub at level of 40% in Bersin hay dates in late pregnancy at postpartum improved the productivity and the reproductive performance of she camels. It improves the postpartum first ostrich interval, number of services, conception rates, days open, pregnancy rates, duration of placental drop, calving interval, and growth performance of their calves. Now I move to another effect. It's photoperiod. If you remember, I mentioned the seasonality, the nutrition, and now I'm moving to the effects of photoperiod. Historically, in the past, the season of reproduction in camels was attributed primarily to nutrition factors and temperature. But now, recent findings show that photoperiod influence most aspects of reproduction in both male and female camels. This is the work done by Swilem 2008 and all in 2018. And blend folds were used to shorten the daily photoperiod for 6 dromedary camels bulls, which resulted in improving reproductive performance. From this work, I took this table and like you see that, for example, the testicular volume increased after this manipulation of photoperiod, and all the other parameters are increasing and improving after this treatment. Coming back now to the pregnancy losses, which is another indicator in this productivity performance. We see from Nagy to it's his presentation recently in 2024. I have to emphasize that when Nagy presents or writes something, you have to keep in mind that he is dealing with intensive system and not extensive system. Then he reported that total loss in dromedaries reaches 25%, starting from 13 days of gestation. 2/3 of embryo and fetal losses occur within the first 60 to 90 days without any visible signs. The remaining one, this is why normally when you are asking a herder, he said to you that they give low fertility rates because they didn't know mainly the embryonic mortality, they didn't notice them, then they give you a result which is not correct. Remaining 1/3 results in either abortion or perinatal mortality. 10% of pregnancies beyond 60 days are lost, while 90% results in the birth of healthy camel calves surviving more than 48 hours. Under intensive management, the abortion rate was 5.1% and prenatal mortality was 4.1. From yesterday, we learned that these rates of mortality are very high. Why? Because normally we collect them from very extensive herds, while this camelicious really farm is very high intensive way to deal with. This is why you see here that these rates are very low. Now, I would like to go now to the advances on reproduction manipulation. We learn now a lot from our friends. I'm sure that the presenters after me, they will give a detail. I will now go to these details because I'm working since 2003 on reproduction physiology and reproduction management, mainly artificial insemination. Then I will summarize my opinion about these advances. For camel, I have to emphasize that there is now really, we still have this intensive dominating. We don't have any farm with intensive one, which make these advances difficult to implement. But learning from others, we will really start here in ASARI. We will really implement some techniques and some research that will advance the management and the dealing with these challenges in reproduction. There is, for example, for artificial insemination, what I would like to tell you that for me, artificial insemination is a package. It's not technique, because if we are judging just technique for one result, the fertility rates, for example, or the birth of camels, we will really suffer from all the. I will skip some. What I would like to say is we learned a lot from our friends, and we will start from their successful stories. For example, for protocol of synchronization, we know that using GNRH and using PGF2 alpha can improve, for example, the protocol of synchronization. Using to avoid this problem, I think Lulu Skidmore, Professor Lulu, she will really puts a lot of emphasis on this. I will just tell you that, for example, here in ASARI, we will try to start from, for example, to liquefy the semen, to use directly the enzyme from papain, or instead of trying other ways, we wish to start from their really nice results instead of coming back and repeating. Unfortunately, perhaps she will give us more details on the outcome of artificial insemination, but I know in my knowledge, I know that there is few limited numbers inseminated because of this high problem of making the semen very liquid. Then we have a few fertility trials. We will start, for example, I know that she will put more emphasis on this. For example, to collect semen, we have to train a lot males, but here, if we have to start this technique, we will really use this technique of condom to directly collect the semen. We have other challenges, for example, poor repeatability result, the fresh shelled semen. I'm working on the semen from ram and from goats for many years ago, 20 years ago. I have the experience. Then I'm changing temperature, I'm changing extenders, I'm changing, anytime I change one component, the result will change. I think the same will be after liquefying this semen, it will be like other semen. It's with less concentration, but I know that spermatozoa is the same for many species. For the freezing also has some problems because she will perhaps talk about that. For the freezing, the spermatozoa are sensitive to heat stress, to temperature shock. Then we will deal with some, but at this stage, and I think we are far from that. Embryo transfer, I think also that we still very late, far from that, because really from NYC we learned that the success story pregnancy rate was 19 to 40 many years ago, but today here they reported 20 to 92%, the winning rate 27. But in my opinion, this really technique should be used, for example, for racing animals. But today here in Morocco, I think we are far to use this technique. Strategies starting from what direction? I would like to share with you some, especially with the director, Sequisni, I would like to share with you this point. Implementing a good selection genetic protocol but without copying the model of cows, because you know here in Morocco we lost a lot of diversity. Then we don't hope to repeat the same mistake. We already have made mistakes in the cattle herds. Then we are aware that normally we keep eyes open on diversity and other things, resilience. Implement an experimental farm, because you know that normally physiology and others should be really studied in farm animals. Encourage intensive farms. You see here that Moroccans started to implement cooperatives and good farms, starting from successful protocols achieved by others. I told you that normally we will gather all the success stories from the previous studies to implement them, study the physiology of our population, generate a package of techniques, because for me, artificial insemination and either embryo transfer is not just one technique, it's a package of techniques. We have together the male side and the female side, a good plan of capacity building. We have to implement a good plan of capacity building, and without really forgetting sustainability focus, maintaining and supporting the extensive system. This means that when I tell you that we have to implement intensive, we have to add artificial insemination and we are not killing the extensive system. The extensive system is dominating in Morocco, and we will get it until the end of this earth, I think. Future perspectives. If we take camel milk production as an example, we have to focus on genetic improvements, implement enhanced breeding programs using assisted reproduction technologies, the technology integration, adopt smart farming techniques and data analytics to optimize operations, product diversification, develop value-added camel milk product to increase, and meats, I'm sorry, I forget, to increase market opportunities, sustainable focus, emphasize environmentally friendly products method to ensure long-term sustainability. This is the end of my presentation. Thank you for your attention.
Thank you very much, Dr. Al-Ameri, for this insightful overview of the production performance in Morocco and beyond, and for the technologies of reproduction. I can open stage for one question. So if you have any question, please raise your hand. Yes.
Thank you very much, Professor, for this very interesting talk. And I was intrigued to see on your last slide that you want to also select for increased diversity and don't repeat mistakes happened in the past for the cattle. So what other types of traits are you going to select for except from high productivity? Do you know already?
I have this answer to this question, but the mistakes we made, for example, in cattle, we really imported a lot of Holsteins, and now we still have own cattle. For example, we still have just one breed, which is UM6P breed, and even this one we have just 5,000 heads. It's a pity because we have 3 million. And among this 3 million, we have just 5,000. It's a pity. But I think with breed, we are really aware about don't losing this biodiversity and not copying from this example. This is why, but I think he can give more insights. Thank you very much for your question.
Thank you very much, Dr. Al-Ameri. So now we will move to Professor Abdelmalek Sraoui, that will tell us about physiological basis for an optimal management of camel production in the Moroccan context. Professor Abdelmalek Sraoui, welcome, and you have the floor. Please go ahead.
Thank you very much. Bismillah ar-Rahman ar-Rahim.
بسم الله الرحمن الرحيم. شكرا للأستاذ العمري على هذا العرض اللي قدمتي. بالنسبة لنا احنا في الأقاليم الجنوبية تحظى الإبل بمكانة هامة عند أهل الصحراء في الأقاليم الجنوبية، فهي جزء من تراثهم العريق وتعد مصدرا للرزق ورمزا للفخر والوجاهة ووسيلة للتنقل والترحال. وعلى مستوى التكاثر وزيادة في الإنتاج هناك طفيليات داخلية تكون في مبهل الناقة وتكون على مستوى لسان الناقة. هذه الطفيليات الداخلية تمنع التكاثر. نحن الكسابة على مستوى الطب التقليدي نتدخل في الصباح الباكر بعد طلوع الشمس ب10 دقائق ونقوم بإزالة طفيليات داخلية. يكون الطبيب التقليدي له تقنيات خاصة بالنسبة للتكاثر. بالنسبة للتكاثر هناك حشرة عبارة عن دودة. هذه الدودة تنشط في فصل الصيف وتكون نائمة على أغصان الطلح وتأكلها الناقة. النوق اللي في حالة الحمل، في حالة التقريب احنا باللهجة المحلية. هذه الدودة وهذه الحشرة تسبب في الإجهاض المبكر، في الإجهاض وتسبب في إعاقة الفصيل. سؤال هو هل هذه الباحثين وهذه المختبرات التقنية والعالمية وصلت والله عندها أبحاث بالنسبة لهذه الحشرة باش يعني عنها بالنسبة هي تكون في شجرة طلح؟ وشكرا.
شكرا الأخ الكريم على السؤال ديالك. الشطر الأول من السؤال ديالك أنا متفقة معاك، وهذاك الشيء علاش وريت من تحديات التكاثر كاين التحدي ديال الأمراض، لأن ما يمكنش اليوم حنا نجيو نقولوا نديروا تلقيح اصطناعي ونديروا نقل الأجنة في حيوانات هي كتشكي من بعض الأمراض. هذه الأولى. الثانية، أنا سمعت من بزاف ديال الكسابة هذه الدودة ديال الطلح، ووعدتهم أننا هنا في أصاري غادي نحاولوا أننا نشتغلوا عليها، غادي نخرجوا حنا وياكم عدة مرات إن شاء الله ونشتغلوا عليها ونعرفوها شنو هو المشكل. وكنظن الأستاذ كيشو راه كاين معنا هنا في القاعة، هو غادي يتكلم على الأمراض وغادي تبان لكم راه هي عائق، لأن الأمراض هو التسيير بجميع المراحل ديالو، التغذية، الصحة، كلهم هاد المكونات هما ضروريين باش نمشيو لقدام بالإنتاجية. وهذا الشيء اللي كيعاني منه اليوم الإبل راه هو اللي كانت كتعاني منه الأغنام عندنا في المغرب. إلا شفتوا الدراسات اللي كانت مديرة قبل من السبعينات على الأغنام في المغرب غادي تلاحظ بأن كانت عدد الأموات في هذيك المناطق اللي فيها الثلج ولا هذه كتمشي 30%، كتمشي 50%، كيمشي كيضيع كل القطيع. ولكن الحمد لله اليوم بتضافر الجهود ديال المربي مع البحث العلمي مع بزاف ديال المؤسسات اللي كتشتغل على هذا الشيء، الحمد لله ولينا حنا كنقولوا لهم ما خصناش نفوتوا 40%، 4%. وهذاك هو الرقم اللي شفتوني كنت كنقدموا على هذيك كاميليشيوس اللي هي وصلت لواحد التقدم ملحوظ، واللي كتبين بان بتضافر هذيك الجهود ما بقينا ان شاء الله. حنا راه يلاه بدينا، ان شاء الله غادي نضافروا الجهود ديالنا جميعا ونمشيو في هذا الاتجاه. شكرا لك.
Thank you very much, Dr. Lamé. Please, Professor ASARI, you have the floor.
بسم الله الرحمن الرحيم والصلاة والسلام على أشرف المرسلين. السلام عليكم. Bonjour, good morning everybody. I will talk in French. La thématique que je voudrais aborder, c'est relative aux bases physiologiques pour une gestion optimale de la reproduction dans les élevages extensifs au Maroc. Je voudrais partager avec vous mon expérience ici dans Laayoune et dans les régions sahariennes que j'ai démarré en 1986. Et cette expérience, je suis le vétérinaire, je voudrais juste me présenter. Je suis docteur vétérinaire et professeur à l'IAB Hassan II. Je suis spécialiste de la reproduction, mais de la reproduction comparée. Je ne fais pas uniquement le dromadaire, mais je fais aussi la vache laitière, enfin la vache, les ruminants, le cheval et les animaux de compagnie. Donc ça me donne, c'est une voie que j'ai choisie qui est difficile parce que je ne me suis pas concentré sur une seule espèce, mais ça me permet d'avoir une vision globale sur la reproduction chez les différentes espèces pour que je puisse avoir une conduite face aux différents problèmes qui peuvent se poser chez ces espèces domestiques. Suivant, s'il vous plaît. Ah bon? Voilà. C'est bon. Alors, nous avons fait beaucoup de choses pour le dromadaire. Alors, le dromadaire, il est considéré par les chercheurs scientifiques comme un matériel biologique extrêmement important depuis de nombreuses années, parce que cet animal représente des particularités par rapport aux autres espèces qui concernent l'anatomie, qui concernent la physiologie, qui concernent la reproduction, qui concernent la génétique, qui concernent l'immunité et qui concernent la pathologie. Et chaque année, on découvre des choses particulières chez le dromadaire qui n'existent pas chez les autres espèces dont nous sommes habitués. Par contre, cette espèce n'est plus considérée comme un animal exotique, curieux, que l'on a hâte d'aller voir le dromadaire. Maintenant, le dromadaire est passé dans nos habitudes, même dans notre champ visuel, puisque le dromadaire n'est pas uniquement dans les zones sahariennes, il est aussi dans le nord, on le retrouve à Tanger, sur les plages. Donc même les enfants, maintenant, vous leur parlez de dromadaire, il n'y a aucun problème de curiosité, même en Europe, aux États-Unis, en Asie, partout. Le dromadaire, c'est un animal comme les autres maintenant. Donc, il faut le considérer de cette façon-là pour qu'on puisse savoir son potentiel de production. On dit que le dromadaire ne produit pas beaucoup, mais il ne produit pas beaucoup dans le biotope saharien. C'est normal. Il y a la sécheresse, il n'y a pas d'eau, Quand vous transposez le dromadaire, vous le mettez dans des conditions meilleures, il commence à produire. C'est pour ça que le dromadaire aussi, il est soumis aux règles de production, il répond à ces règles de production. Nous avons des dromadaires qui produisent du lait, bien entendu, et que ce soit une production légèrement, j'allais dire, traditionnelle, comme une production de façon industrielle. Donc ça, c'est aussi important, comme le dromadaire produit de la viande. On peut produire de la viande de façon traditionnelle, mais si on soumet le dromadaire à des conditions intensives, des feedlots par exemple, le dromadaire va répondre. Donc le dromadaire va s'habituer, va s'adapter à son biotope. Donc cette idée, toujours transposée, le dromadaire ne produit pas, la reproduction, elle est lente chez le dromadaire, elle est longue chez le dromadaire, mais ça dépend dans quel sens on le comprend. Alors malheureusement, sur le terrain, on a noté que la structure de l'élevage du dromadaire et de la gestion de l'élevage du dromadaire ont été délaissées. La structure de l'élevage, je parle du troupeau, parce que depuis hier, on parlait de beaucoup de facteurs qui handicapent la production et la productivité chez le dromadaire, mais depuis 1980 à nos jours, il y a eu beaucoup d'avancées. La première avancée, c'est que les éleveurs se sont organisés en association, en coopérative et en fédération. La preuve, hier, on avait un panel où il y avait plusieurs représentants des associations, chose qu'on ne connaissait pas auparavant. Deuxième chose, c'est que le lait a été complètement, il a dépassé, j'allais dire, les habitudes culturelles et les habitudes ethniques pour rentrer sur le marché, et il a commencé à être commercialisé depuis une trentaine d'années, chose qu'on ne pouvait pas croire, que même les éleveurs, même les ethnies, même les gens du Sahara ne voulaient pas entendre parler de vendre le lait. Alors que ce produit a été mis sur le marché malgré toutes les difficultés depuis une trentaine d'années, que ce soit localement, c'est-à-dire dans la région saharienne, puis il a dépassé les frontières et le territoire saharien pour aller dans le nord. Là, on va voir beaucoup d'élevages périurbains qui produisent du lait. Et puis sur l'international, il y a eu du lait qui a été exporté ailleurs. Donc, après quelques années de cet effort, de ce pas en avant justement pour commercialiser le lait, l'éleveur s'est rendu compte que le lait représentait une trésorerie pour lui. Il gagnait de l'argent, chose qu'il ne pouvait pas faire auparavant. Donc il a commencé à gagner de l'argent et ça représente une trésorerie. Maintenant, nous parlons de chaîne de valeur lait, chaîne de valeur viande. C'est grâce justement à ce pas en avant que les producteurs ont osé mettre le lait sur le marché. Donc, cette immersion sur le marché et dans le domaine de la commercialisation, il a commencé timidement, mais il a pris son envol et maintenant, on est fiers que le lait de dromadaire soit commercialisé. On est fiers que nous, qui ne sommes pas de ces régions-là, qu'on ait le lait chez nous, qu'on le consomme, mais le lait, il a des vertus médicinales et diététiques que sont pris. Hier, les collègues au niveau du panel, ils ont parlé de beaucoup de problèmes. Comment commercialiser le lait? Comment on produit du lait ici, une quantité importante, mais qu'on n'arrive pas à acheminer au nord. Ça, c'est un problème. Et ceci a donné la possibilité à ces éleveurs, certains qui viennent de ces régions-là, et qui ont mis en place des petits troupeaux autour des villes. Et vous avez certainement constaté ces élevages périurbains au niveau de Rabat, au niveau de Oujda, au niveau de Tanger, au niveau de Fès, au niveau de Marrakech, au niveau d'Agadir. Partout, il y a des petits élevages périurbains qui peuvent approvisionner la population ou le consommateur local pour des raisons certainement beaucoup plus médicinales que des raisons d'aliments de base. Donc, le lait a été, ce pas de commercialisation a poussé d'abord les chercheurs, parce que la recherche a été faite au niveau de l'IAB Hassan II sur la pasteurisation, donc la pasteurisation du lait. Et le fait d'avoir pasteurisé le lait, ceci a permis de le conserver et de lui donner un peu plus de temps pour qu'il soit conservé. Alors, il y a plusieurs types de lait. Là, on a parlé hier à Dakhla, à Laayoune. Maintenant, nos amis qui sont à Laayoune, nos amis qui sont à Dakhla, qui produisent du lait. Mais nous avons parmi nous une enseignante de l'IAB Hassan II qui a aussi mis au point un lait, ce que vous voyez, la petite bouteille qui est à droite avec le fromage. Et ceci a été réalisé dans les coopératives laitières de Laayoune, de Dakhla, de Smara, de Glmim. La même chose pour la viande. Là, je vais juste dire un mot sur la viande, parce que ce n'est pas mon topo. Donc la viande a aussi connu une valorisation du fait que la viande est produite de façon importante. Et tout ce que vous voyez comme produit, sous-produit, valorisation de la viande, ça a été réalisé aussi au Maroc par notre collègue et confrère, le professeur Daoudi. Parmi les autres avancées, c'est qu'il y a eu l'organisation des courses, l'organisation des concours, la participation à des salons, aux foires. Là, on a entendu hier que beaucoup de nos éleveurs partaient régulièrement au salon de Meknès pour présenter leurs produits. Et ça, c'est aussi une preuve que les gens ont commencé à connaître et à apprécier justement le produit. L'intervention aussi de l'État, il ne faut pas l'oublier, l'intervention de l'État et de ces structures pour développer le dromadaire dans les régions de Glmim, de Laayoune, de Dakhla, de Smara, de Boujdor, Assazak et toutes les régions où le dromadaire existe. Alors sur le plan scientifique, les avancées qui ont eu lieu, c'est que il y avait 3 grands congrès internationaux, celui de 1999 à Ouarzazate, celui de 2018, c'est l'Isocard à Laayoune, et actuellement, ce que nous sommes en train de vivre aujourd'hui, c'est l'International Conference of Camelids ici à Laayoune. Donc des conférences à l'échelle nationale. On a sillonné toutes ces régions-là pour présenter nos travaux de recherche, pour former et sensibiliser les éleveurs, former les vétérinaires, former les ingénieurs, former les techniciens à travers plusieurs séances à partir de 1995. Et aussi, il y a eu beaucoup de grands séminaires locaux qui ont été réalisés en 1987 et en 1987. Alors, sur le plan recherche, bien entendu, la recherche, c'est le parent pauvre des avancées scientifiques. Mais malgré ça, il y a eu des moyens financiers, modestes soient-ils, irréguliers soient-ils, ils ont donné un peu de progrès, justement, à faire avancer la science. Et nous comptons beaucoup, justement, sur ASARI et sur l'Université Mohamed VI pour donner encore plus de moyens, parce qu'à la fin, je vais dire une autre idée sur ce point-là. Alors, de toutes ces réalisations, bien entendu, la structure de l'élevage n'a pas changé, malheureusement. L'éleveur, avec ses animaux, est resté le même. La gestion est restée la même. Donc, c'est ça ce qui nous donne des problèmes pour aller récupérer des données fiables. Nous avons un problème pour récupérer des données fiables parce qu'il n'y a pas d'enregistrement. Il n'y a que l'oral. Et ma chamelle, elle a mis un nom. Quel mois? Je ne sais pas. Combien de chamelons vous avez? Quel est le taux de mortalité? Tout ça, ça reste des suppositions. Et nous en avons vu hier, à partir des différentes présentations, que le problème de données fiables ne peut pas nous permettre de présenter des données à risque, des données qui ne sont pas fiables aussi. Donc, si on veut travailler dans l'objectivité, nous avons besoin de développer un système pour donner à l'éleveur, permettre à l'éleveur justement d'archiver ces données au sein de l'élevage, combien elles sont importantes, que ce soit pour le... Ça, c'est quelque chose qu'on demande aux éleveurs de bovins, aux éleveurs de chevaux et aux éleveurs de petits ruminants. Et là, on se heurte aussi à ces problèmes-là. Donc, à mon avis, on a fait beaucoup de choses, mais la structure de l'élevage n'a pas été touchée et les éleveurs ici présents peuvent en témoigner. On est resté la même, c'est toujours la même. C'est des animaux transhumants, c'est des animaux qui se déplacent à la recherche des sources hydriques, à la recherche du meilleur pâturage, etc. Donc ça, c'est un point qu'il faudrait mettre en avant. Donc, mais on a la possibilité, avons-nous la possibilité de créer des fermes, des espèces de fermes avec des programmes de biosécurité ciblés contre certaines maladies pour vacciner les jeunes, pour faire des plans de supplémentation de sélénium. Le sélénium a été démontré par Werner en 2014 comme c'est un facteur d'immunité important parce que les animaux qui ne sont pas supplémentés par le sélénium, ils connaissent beaucoup de problèmes dans leur vie. Voilà, donc ça c'est en guise d'introduction. Là, ce que je voudrais partager avec vous, c'est un travail que j'ai réalisé, cette fois-ci dans des troupeaux structurés. Ce sont les troupeaux de militaires qui nous ont rendu un grand service et auprès de qui nous avons beaucoup appris. Parce que tout est archivé. Toutes les données sont là, correctes. Des fois, il y a une petite contradiction, mais on peut les noter. Mais ce n'est pas quand on arrive dans un élevage où tout c'est l'oral, il n'y a rien d'écrit, c'est à peu près, etc. Donc, ceci ne va pas rendre service ni aux vétérinaires, ni aux techniciens, ni aux responsables, ni à l'ingénieur, ni à la personne qui voudrait mettre en place un travail de recherche correct. Donc, ce travail est le fruit d'un suivi régulier de troupeaux camelins avec des enquêtes auprès des éleveurs de la région. On a identifié les caractéristiques du dromadaire dans son berceau afin de ressortir les facteurs, justement, et pouvant constituer une contrainte au développement de l'amélioration de son élevage. Donc, la méthodologie, elle est un peu longue, mais je voudrais juste vous citer à peu près le nombre d'effectifs que nous avons touchés. Le nombre pendant 7 ans, nous étions présents au niveau de ces troupeaux pour pouvoir récupérer l'information. Ce sont des informations qui vont concerner aussi le mode de conduite, les déplacements des animaux, l'alimentation, l'appréciation de l'état des animaux, les informations individuelles qui concernent l'identification de l'animal. C'est un problème aussi, l'identification, et l'État essaie de faire un effort pour que tous les animaux soient identifiés afin de pouvoir les suivre et de rentrer dans le principe de traçabilité, que ce soit des traçabilités individuelles, mais aussi des traçabilités pour connaître les problèmes d'évolution des maladies, surtout les maladies qui viennent des frontières. Pour le dromadaire, ce sont des frontières un peu poreuses, donc l'animal rentre et il rentre chargé avec beaucoup de pathologies. Le signalement de ce dromadaire, le sexe, le type, il n'y a pas de race, mais on parle de le type, il n'y a pas de race. Et hier, on a entendu parler par des collègues comme quoi ils insistent sur le type. Peut-être les généticiens ici présents, nous en avons à peu près 3 généticiens de renommée internationale qui peuvent aussi nous dire quand est-ce qu'on va commencer à parler de race de dromadaire. La date de naissance, l'état sanitaire, etc. Tout ça, ce sont des données importantes à mettre en place. Et puis, nous avons pu calculer l'âge de mise à la reproduction, l'âge à la première mise-bas, le taux de fécondité, la distribution des saisonnières des mises-bas, l'intervalle des mises-bas et le taux de mortalité. Alors, le taux de mortalité, j'ai mis des points d'interrogation parce que j'ai un chiffre affolant, c'est celui de 58%, mais le taux de mortalité des jeunes, en 1997, il était désastreux parce qu'il y avait des diarrhées néonatales et puis il y avait des intoxications de femelles pendant la période du postpartum qui ont causé justement des problèmes de mortalité. Alors, je vais commencer par l'âge au premier chamelage. Ça, c'est un chiffre qui nous intéresse pour savoir à quel moment la jeune femelle va rentrer en reproduction, pourrait rentrer en reproduction, pour être productive. Pas rentrer en production, pour être productive. Donc, selon les différents travaux que nous avons menés, regardez sur le graphe de gauche, c'est à partir de 2 ans à 2,7 ans qu'on a pu noter une naissance. Ce n'est pas un chiffre faux, c'est un chiffre vrai parce que ça représente une petite population. Donc, c'est vrai que le potentiel, la femelle peut devenir pubère très précocement. Alors, dans toute la littérature, on dit que la puberté est atteinte à 4 ans, à 5 ans, que l'animal ne peut mettre qu'à partir de 6 ans, etc., etc. Mais quand on fait le suivi, quand on est présent, quand on a des données fiables, on peut construire quelque chose d'objectif. Donc, sur les différentes années, donc la moyenne, c'est de 4,12 à 0,7 ans, bien sûr. La variation est de 33 à 71 mois. Et nous avons identifié 3 populations. Il y a les femelles qui sont précoces. Alors, ils sont précoces parce que certainement, ils sont nés, ces femelles-là, ils sont nés très précocement au début de la saison. Ils ne sont pas nés à la fin de la saison, vers le mois d'avril et vers le mois de mai. Quand les femelles naissent au mois d'octobre, on a la chance justement que ces femelles-là puissent devenir pubères très, très, très jeunes. Alors, les femelles qui sont tardives, sont que l'on voit sur la droite de ce graphe-là, eh bien, ce sont des femelles, soit ils avaient des problèmes de reproduction qui ne sont pas identifiés, bien entendu. Le problème, c'est qu'il faut être là pour diagnostiquer, pour vérifier combien de fois j'assistais à un traitement empirique de l'infertilité chez la chamelle du dromadaire. On prenait du sel, le sel gemme, qu'on concassait et on va le déposer au niveau de l'utérus. Ça, c'était le moyen, peut-être ça marchait. Ça, c'est une médication empirique. En tout cas, ce n'est pas basé sur la science, mais elle est basée sur l'observation. Et les autres femelles qui représentent 56% qui ont un âge entre 44 et 45 mois pour mettre la première fois. Ce tableau aussi nous donne un peu l'âge au premier chamelage dans les différents pays, que ce soit asiatique et africain, vous voyez, pour un peu comparer nos données par rapport à ce qui est trouvé ailleurs. Alors l'âge à la première saillie fécondante avec possibilité de gestation. Oh. Je voudrais dire un mot sur la saisonnalité. La reproduction chez le dromadaire est jusqu'à présent saisonnière. Mais il y a beaucoup de facteurs qui influencent justement cette saisonnalité, notamment la nutrition. Tout le monde, depuis hier, on parlait de la nutrition. Rafat, hier, il a beaucoup discuté la nutrition chez le dromadaire. Le panel, ils ont beaucoup discuté la nutrition. Les exposés et les communications de l'après-midi ont focusé sur la nutrition. Donc la nutrition, c'est un élément important. Et regardez la distribution des naissances, le pic est au mois de février. Et on peut déjà commencer au mois d'octobre. Octobre, on a les premières misbas qui vont commencer à arriver. L'intervalle entre misbas, il est extrêmement important parce qu'il va déterminer la productivité. La femelle qui est présente dans le troupeau, il va produire combien de chamelots, on va dire, par an? Mais c'est impossible par an puisque la durée de gestation est de 12,5 mois à 13 mois. Vous voyez, donc, on détermine justement cette productivité, elle est importante. Donc, l'intervalle chamelage, il est important. Et la moyenne, elle est entre 24,03, mais il faut le prendre avec beaucoup de précaution parce que volontairement, l'éleveur ne souhaite pas mettre sa femelle à la reproduction après la mise-bas. Non, parce qu'il voudrait conserver une partie de la production du lait aux jeunes chamelons pour qu'ils puissent survivre. Bon, allez, je vais passer parce qu'il me reste 5 minutes. La distribution globale de l'intervalle mise-bas, ça y est, fécondante, est en moyenne de 11,8,2 mois, mais ceci, c'est le postpartum. Alors, il y a certaines femelles, à l'image de la jument, là, le dromadaire peut, dans les 7 jours qui suivent, peut être gestant, parce que ceci dépend du placenta. Le placenta, il est épithéliocorial, la délivrance se fait dans les 3 heures, et la femelle retrouve facilement sa reproduction dans la semaine, sinon dans le mois. Par contre, si on laisse la femelle allaiter son jeune, on va développer un anestrus postpartum sous l'effet de la production du lait. Bon, allez, le taux de reproduction. Deux petites comparaisons entre le système nomade extensif, dont le taux de reproduction est de 20 à 50%, celui de l'intensif est de 50 à 60%. On imagine que l'intensif, la nutrition est prise en considération. Le taux de mise bas est de 39 à 60% parce que le taux de mise bas, c'est vraiment l'indicateur objectif de la productivité. Ce n'est pas le taux de gestation, parce que la gestation, il peut être perdu à n'importe quel moment pendant son évolution. Mais ce que nous jugeons objectif, c'est le chamelon qui est né. Après, pour le système intensif, c'est entre 55 et 80%. Le taux de perte de gestation est de 3 à 33% et de 2 à 5% pour le système intensif. Mais aussi, encore une fois, ici, nous ne pouvons pas, dans les conditions de terrain, évaluer le taux de mortalité embryonnaire. Et le taux de mortalité embryonnaire, il est énorme dans les élevages camelins parce qu'on n'a pas moyen de faire un suivi échographique de la gestation. Et puis les mortalités, regardez, entre 7 à 25%, ça peut aller jusqu'à 90%, jusqu'à la dissémination. Je me rappelle ici quelques années, l'éleveur, il avait 50 dromadaires. Sur les 50 qui ont mis bas, il n'y avait que 10 qui ont pu sauver leur chamelon. Les autres sont morts pour des problèmes de parasitisme, pour des problèmes de déshydratation, pour des problèmes d'immunité passive. Alors, quelles sont les avancées maintenant majeures de la reproduction? Tout à l'heure, Madame Bouchra Al-Amiri avait parlé de la saisonnalité et de la mélatonine. Un des travaux qui a été réalisé à l'IAV par Al-Lali a noté qu'avec la mélatonine, on peut introduire un degré d'allongement de cette saison. Et nous souhaitons que la saison soit un peu allongée. Et cette mélatonine peut aussi allonger la reproduction chez le mâle et la femelle. D'autres avancées en matière de reproduction, ce sont des modèles cliniques et endocrinologiques. Comment on peut aborder cliniquement un problème d'infertilité? La caractérisation des hormones, le mécanisme d'ovulation grâce aux travaux de NOSC et de Marie en 1987 qui ont un peu dévoilé ce phénomène d'ovulation et cette ovulation provoquée. Vraiment, c'est des travaux phares qui ont mis maintenant en place. Toute la reproduction a été basée justement sur ces travaux-là. Le contrôle du cycle, la saisonnalité, l'effet de la nutrition sur la fonction ovarienne et l'infertilité. D'autres travaux sur la dynamique folliculaire. Bien entendu, on a résolu le problème de l'ovulation, mais on n'a pas résolu le problème de la dynamique folliculaire. Comment ces follicules, ces cohortes de follicules qui existent sur les ovaires, on va les suivre pour avoir un follicule préovulatoire, et ceci grâce aux travaux de Skidmore en 1996 et Tibaré NOSC en 1996, qui ont un peu décrit justement cette dynamique folliculaire, mais ceci grâce au suivi échographique, bien entendu. Et là, on a pu déterminer la sélection, le recrutement, la croissance, la maturation et la régression du follicule, jusqu'à la régression du follicule. Malheureusement, chez le dromadaire, certains follicules ne régressent pas et continuent toujours à croître jusqu'à devenir pathologiques. Et ça, ceci touche à peu près 30% des femelles. Les facteurs physiologiques qui vont affecter la dynamique folliculaire, c'est la saisonnalité. L'effet de la taille folliculaire, il a un effet sur la qualité de l'ovocyte. La nutrition, encore une fois la nutrition. L'effet de sur la croissance folliculaire, l'effet sur la qualité de l'ovocyte, la gestation aussi, l'effet négatif sur la dynamique folliculaire, parce que pendant la gestation sur le dromadaire, nous avons un corps jaune dominant qui doit démarrer de la mise en place de la gestation jusqu'à la fin. Donc, il va inhiber toutes les structures folliculaires qui vont pouvoir se mettre en place. Et puis, la lactation qui est un problème aussi dans la nostrus, parce qu'il va avoir un effet négatif sur la croissance folliculaire et il va avoir un effet négatif sur le corps jaune. Alors, d'autres avancées majeures hein? Sur l'infertilité. Alors, l'infertilité, c'est un problème. Un troupeau où il y a des femelles qui ne se reproduisent pas, c'est un casse-tête pour l'éleveur, parce que c'est lui le premier concerné. Sa femelle, elle est là, on va dire qu'elle est à l'hôtel, il mange, il s'alimente, mais il ne produit rien. C'est aussi un casse-tête pour le vétérinaire, parce que s'il n'est pas prévenu à temps, c'est un problème, parce que le délai de diagnostic va s'allonger et la thérapeutique risque de ne pas être correcte. Donc, il faut s'appuyer sur l'examen physique, de la notation de l'état corporel, le BCS, c'est important. La distribution des réserves graisseuses sur l'animal sont importantes, et ça, on le voit à l'œil nu. On n'a pas besoin de peser l'animal. La palpation transrectale et l'échographie, la vaginoscopie, la cytologie, la bactériologie de l'endomètre pour évaluer la santé de l'utérus, la biopsie utérine dans certaines situations d'infertilité, l'hystéroscopie, la cytogénétique, parce que ça, c'est un problème dans nos élevages, parce qu'on ne renouvelle pas le géniteur fréquemment. Le géniteur, il est là pendant 4 ans, des fois 5 ans, et il commence à saillir la première population des mères, mais il commence à saillir aussi ses filles. Et ça pose un problème de consanguinité, aussi l'endocrinologie. Certaines maladies qui vont causer l'infertilité, les endométrites, infections, les endométrioses, les adhérences, une pathologie que vous voyez là en bas, c'est l'adhérence de la bourse ovarique, et ça n'existe que chez le dromadaire, et les traitements principalement calqués sur les autres espèces avec différents ajustements. Le problème aussi, c'est les avortements. On n'a pas de chiffres. Celui qui parle d'avortement, on n'a pas de chiffres. À peu près, les avortements. Parce qu'il y a des avortements qui se font sur les parents qu'on n'arrive pas à évaluer. Mais on peut aussi faire des tests de trypanosomose, des tests de brucellose qu'on ne connaît pas aussi l'état sanitaire du cheptel marocain en termes de brucellose. La fièvre Q. On ne connaît pas d'autres éléments qui font défaut et qui peuvent être la cause justement de ces avortements. Ce sont les carences en oligo-éléments, les mortalités immunitaires, l'immunité passive. Cette immunité, c'est juste après la mise bas. Si la femelle ne produit pas suffisamment de colostrum, le chamelan risque de ne pas survivre longtemps. Et puis, il faut qu'on fasse une immunisation des mères. Le mâle, je vais dire un mot parce que c'est un peu le parent pauvre dans un troupeau. On voit plus les femelles, on focus beaucoup plus sur les femelles et on oublie un peu le mâle. Le mâle, on n'a pas aussi de données sur la relation entre l'évaluation clinique et la production de spermatozoïdes et la fertilité. Nous avons un manque d'informations sur la durée de la spermatogénèse, sur le transport des spermatozoïdes et la production de spermatozoïdes. Nous avons aussi des informations clés sur une bonne gestion des mâles reproducteurs. On n'a pas une gestion assez affinée.
C'est vrai. Merci de conclure parce qu'on a largement dépassé le temps.
Alors, je vais conclure avec cette diapositive-là pour parler des performances de reproduction. Donc, l'objectif ultime de tout élevage, tout système de production, est de sevrer, comme j'ai dit tout à l'heure, un nouveau-né en bonne santé. Il faut que la gestation soit à terme, que la naissance se déroule dans des meilleures conditions et qu'on récupère justement le chamelan. Les performances reproductives du troupeau est déterminé par le taux de gestation, c'est important. Le taux de mise bas, qui est encore beaucoup plus important. Le taux de sevrage, combien de femelles sont sevrées dans un élevage de 100 chamelles. Le taux de fertilité annuel, qui a été entre 12 à 85%. Il y a beaucoup d'auteurs qui ont travaillé sur ce problème-là. Et pourquoi c'est une telle variation ? parce que ceci dépend du système de production, ceci dépend de la gestion de l'élevage, ceci dépend des soins vétérinaires, ceci dépend de la nutrition. Et les dromadaires, on va se poser toujours cette question, est-ce que les dromadaires sont des animaux à faible, justement, efficacité reproductive, ou bien est-ce que c'est nous qui ne comprenons pas ou ne gérons pas suffisamment la reproduction chez le dromadaire? Et je vous remercie.
Merci beaucoup, professeur Seravi, pour cette présentation. Maintenant, j'ouvre la discussion pour une question peut-être, une seule question pour les gens qui veulent poser une question. Voilà, on a une question ici.
Je remercie mon collègue professeur Saeb Malk pour son exposé. Je dirais que c'est une revue de tout ce qui a été fait sur le dromadaire depuis le temps jusqu'à nos jours. Et je voudrais mettre le point sur deux choses. On a parlé des problèmes de dégénérescence d'embryons, d'infertilité, de problèmes d'avortement, des problèmes de faible productivité. Et ceci, on a essayé de le lier à des problèmes pathologiques et également à des problèmes de nutritionnels. Mais je voudrais maintenant, tout de suite, mettre le point sur la partie nutritionnelle. Quand on parle de nutrition, on oublie une chose qui est très importante dans l'élevage du camelin. C'est l'apport pastoral. C'est un animal qui exploite des territoires immenses dans la région du sud de parcours, avec une multitude d'espèces végétales, depuis la strate herbacée, la strate arbustive à la strate arborée. Donc le camelin exploite toutes ces strates de végétation et donc il fait une ration qui est plus ou moins équilibrée en termes de protéines, etc. Et surtout quand il s'alimente sur les acacias. L'acacia radéana essentiellement qui contient environ 40% de protéines, d'azote non protéique. Deuxièmement, on a également parlé d'un problème de sélénium. Et on sait pertinemment que nos sols, dans ce pays-là, les gens qui ont travaillé, les collègues des sciences des sols qui ont travaillé sur la qualité des sols marocains en matière de minéraux, ils ont constaté qu'il y a une déficience énorme en matière de sélénium. Et ce sélénium, bien sûr, vous savez qu'il provoque des maladies essentiellement, qu'aussi bien chez les camélidés que chez les petits ruminants. Essentiellement, chez les petits ruminants, il cause la maladie, ce qu'on appelle la maladie du muscle blanc. Le muscle blanc, qui est qualifié. Troisième chose, je crois que pour tout développement de l'élevage, quand on parle de l'intensif, on ne peut pas l'assimiler à un élevage de bovins laitiers. Parce que, vous savez, les habitudes alimentaires de la région, comme les habitudes alimentaires d'autres régions de ce pays, qu'est-ce qui se passe? C'est que la viande du camelin et le lait de camelin, ce sont des produits aromatisés. Aromatisés avec quoi? Avec les espèces végétales qu'elles consomment sur le parcours. Et si donc on prive l'animal de récolter, de collecter ces espèces végétales, on va perdre sur la qualité aussi bien de la viande que sur le lait. Et je vous remercie infiniment.
Merci. Ce serait, vous voulez dire quelque chose? C'est bon? Désolé, vraiment, on est hors temps. Mais si c'est... Allez-y, monsieur.
السلام عليكم. ديزولي وصلنا حتى الموضوع ديال الداخله وحليب الابل وبينا نديروا دوزولي ناخذ واحد جوج دقائق ما فيها اشكال. المهم على اي التدخل ديالي في اطار شفت التركيز ديالكم في المساله ديال التلقيح الاصطناعي واش كينصب غير على اسباب تكثر القطاع ولا من اجل تطوير الحليب؟ واش بغينا نديروا داك الشيء اللي كينطبق على الابقار، تربيه الابقار في الستينات والسبعينات هو اللي كينطبق حالا على الابل، بما فيه مثلا البقره كانت كتحلب 6 لتر و7 لتر وصلت ل 30 و50 لتر، بمعنى هذا هو اللي كنطمحوا له. واش كاينين افكار ودراسات يعني في تدخيل الزريعه اللي بغينا نلقحوا بها في هذا التلقيح الاصطناعي باش تطور القطيع ديالنا يتحول الى واحد القطيع اللي هو كيحلب يعني بين 30 لتر كما كيوقع في الدول الخليجيه بدلا ل 6 لتر و7 لتر؟ واش هذه من جهه، والسؤال الثاني للمؤسسة الجامعة محمد السادس ومؤسسة فوس بوكرع، واش ما كيفكروا انهم يخصصوا واحد الوقت واحد الدعم ديال اشهار منتوج حليب الابل من اجل توعيه المستهلك المغربي؟ لان ملي كنمشيو للمعارض الوطنيه كنلقاو الاشكال ديال واحد الجزء ديال الناس انهم ما كيعرفوش كاع علاش كيصلح هذاك الحليب، وكيمشيو كيشروه من عند ناقات ما مبستر، كيمشيو يشرو في مناطق اللي كاسبين فيهم الناس النياق هو ما مبستر يعني غير صحي، وراه الباحثين عارفين هذه المساله، وكيشروه بثمن مرتفع مضوبل بثلاثه ديال المرات على الثمن اللي كنبيعوه به حنا بعد ما كنبذلوا المجهود ونوصلوه للمستهلك، وكيعتقد بانه هذاك الحليب يعني غير حقيقي، خاصو المستهلك يشوفوا من ناقه. لهذا بغينا يعني من مؤسسه فوس بوكرع بتنسيق مع هذه الجامعه انهم يديروا واحد البرنامج او واحد الدراسه ويدعموا القنوات الوطنيه من اجل اشهار هذا المنتوج للتعريف به حتى يقتنع المستهلك المغربي. شكرا.
شكرا. الأخ الكريم، موضوع التلقيح الصناعي سوف ننصت له جميعا من طرف الأستاذة سكيد مورلولو. Il va venir présenter موضوع التلقيح الاصطناعي، حيث كاين التلقيح الاصطناعي وكاين نقل الأجنة، هذو وسائل لتحسين النسل، تحسين النسل من جميع الجوانب، جانب إنتاج اللحوم ولا إنتاج الحليب. سوف ننصت إلى هذا الموضوع. مشكل اللي تطرقتي له هو المشكل ديال التسويق، ياك ديال اعتقد الاخوه لو بانيل البارح كان تكلم على هذه المساله هذه، وكل شيء لاحظ بلا كاين مشكل ديال التسويق، مشكل التسويق كاين، ولكن اعتقد هاد لي فورس فيف، هاد تبارك الله، هاد الناس اللي هنايا وهاد المسؤولين والباحثين والناس اللي ديفلوبور كلهم عندهم نفس الهم ديال هذا التسويق ديال هذا، وان شاء الله سوف يوجد له حل عما قريب ان شاء الله.
Merci beaucoup, Professor ASARI. So let me now welcome Professor Nizar Ahmed Douani, that will tell us about the advances in assisted reproduction in camels. Dr. Ahmed Douani is scientific director of the biotechnology research in Dubai. He worked a lot on reproductive biotechnology, including embryo transfer and even cloning, I think calf cloning, I guess. You are very welcome. You have the stage.
Well, thank you, Dr. Balavi, for the introduction, and good morning, everyone. Yeah, thank you. So today I'll be talking about the acceleration of genetic gain in camelids through the use of reproductive biotechnology techniques. But the first question, asked yesterday by one of the delegates was, why reproductive biotechnology techniques? Why do we need them? So if we see by 2045, the world population is estimated to reach about 9 point something billion, and we need a lot of animal proteins to be doubled, particularly in the developing countries, in African countries. So 70% of this increase is expected to come by use of technology to improve the animal production. And the population increase, if we see the population increase, particularly in Africa, is expected to be more than 200% by 2050. So we need these technologies for a lot of the production of animal protein to feed our populations. So what type of technologies are we going to use? these are the common technologies we are using for the animal production and animal reproduction. Artificial insemination. I'm not going to talk about artificial insemination because my colleague, Dr. Lolo Sikkinmore, will be exclusively talking on that. In vivo embryo production through multiple ovulation and their transfer to synchronized recipients. I'll go briefly through that. Then in vitro embryo production by either IVF, XA, and somatic cell nuclear transfer, which we call cloning, and their transfer of those embryos into the synchronized recipients. So we can do also embryo and oocyte cryopreservation and then store them for their future use. Now, the cell and gene banking for future use, if we have some late animals and we want to store their genetic material, we need to have the cell at gene bank, so we can use these cells anytime in the future if something happens to these animals, and we can even produce these animals by cloning now, and we can introduce these animals into new populations for the the diversity of the genetic pool. Then the genetic modifications, where we can use the transgenics for either disease resistance in different areas for different diseases. And we can also use these transgenics for improving their traits. And we can do the knockouts for some like meat production. I'll go directly to multiple ovulation and embryo transfer, which we also called multiple ovulation, collection and transfer of in vivo produced embryos. So in multiple ovulation and embryo transfer, what we need is we have a very superior female, and we have also a very superior male. So we breed this very superior female with the superior male after superstimulation of the female. So instead of getting one embryo, we should get more than one embryo from this female. And we flush these embryos from the uterus of these donors, the good or these superior females, on day 7 to day 8, in between day 7 and 8, and then we transfer those embryos into ordinary females. These ordinary females, which we call as surrogate mothers, are the recipients. They are being synchronized, having the synchronized estrous cycle with the donor animal. We can repeat this process many times in a year. So instead of getting one calf from a superior female, or the combination of a superior male and a superior female, we can have many calves from this combination. Sometimes get stuck. So for this process, what we need to do is we have to do superstimulation of the donors. For superstimulation of the donors, we have the protocols, well-established protocols. I don't want to go through those protocols once again, because everyone, I mean, they are already published. But this one I wanted to go through is, see, we have this is the publication from one of the colleagues, Manjunathan, and he used ECG, equine gonadotropin. So from 1,000 international units to 6,000 international units. What we could see is the 1,000 to 3,000 to 4,000 international units have given better results, and the transferable embryos were better when compared to even 6,000 international units. Same way, he used FSH and compared FSH and ECG. And we could see that transferable embryos were not different when you use FSH or we use ECG only. This is something now which I wanted to talk about, is how to reduce the cost of these hormones, because these hormones are quite costly. And we have been able, this is the publication also from the same group, Manjunathan, and we are using this now in our center as well. We are using hyaluronan in diluting the FSH with hyaluronan. By this method, we have been able to reduce the cost of these hormones to 1/2. So instead of, like, this is the normal protocol where you use 400 mgs of foltropin, this is FSH, for continuously for 4 to 5 days in divided doses, one morning, one evening. So if we use this foltropin with hyaluronan, and also we have used it now alum, which is a conjugate used for vaccine production. So we can dilute these hormones either in hyaluronan or in alum. And if we have seen that we give 2 divided doses, only 2 days. So we reduce the labor cost, and we reduce the stress to animals, and we reduce the cost of the hormones. This giving half of the hormones means 200 mg. Now we call it slow release foltropin with when we dilute it with either this hyaluronan or we dilute it with alum. So we give one shot, 120 mgs, another shot, 80 mgs on day 4 and day 6. That's it. And we see we get all similar results what we get with the control group. So these are the results. We will see that the transferable embryos are about same, whether you give split injection, only 2 injections, 200 mg, or you give 400 mg for continuously for 5 days into 2 divided doses. Same way, this is another hormone, pulsate. I don't want to go through it. This is exactly the same procedure we have been using. And this is also from Manjunath, but we have been using this procedure as well in our center. And we have been able to reduce the cost of these hormones to 1/2. So this embryo transfer technique, like this was super stimulation, and then you need to collect these embryos. From this picture, you can see many of these people you might be knowing are from Morocco. This is from Abdul Haq Anabsi's lab in Abu Dhabi. So this has developed a large-scale commercial embryo transfer programs now in Middle Eastern countries. And after flushing these embryos out of the donors, they need to be transferred to synchronized recipients. So recipients are synchronized. Either we select the recipients by serial ultrasound from the group of camelids, and then inject those who have the mature follicles on serial ultrasonography. We see mature follicles, we inject them with before 24 to 48 hours with GnRH so that they start ovulating. And they are ready for the synchronize it to the donor's estrous cycle. Or what we do is we do treatment of these recipients with progesterone, 100 mg per day for 10 to 15 days. Then followed by administration of 1500 to 2500 international units of ECG, equine chorioanatomic gonotropic, which was also called as PMSG. And this is usually done a day after the donor receives ECG. Or another protocol, we don't use it much, but people have been using is 2 injections of GnRH given 14 days apart, with the second GnRH scheduled to be administered the day after the donor is given or mated. What we have seen in our center is that yesterday what Rafat was talking about, the nutrition is very important. So the body condition of the donor and recipients affects the outcome of this program and embryo transfer quite a bit. So we need to have the donors as well as the recipients in a good body condition score. When we have the good body condition score, it significantly improves the production of the embryos from the donors as well as the pregnancy rate in the recipients. Similarly, we have seen everybody talking about the seasonality in camels. What we have seen is the females they can ovulate whole year. We can stimulate them and they ovulate whole year. The only problem remains with some bulls. Some bulls, we have seen they are ready to mate even in the hot summer months. But when we give them a little bit of controlled weather conditions or a little bit of controlled environment, so they can breed if we put them indoors for some time during the day in hot weather. And we have collected the embryos in in summer months. This was done in June and July, which is quite hot in Dubai. And we got the embryos, we transferred them into recipients, we have pregnancies, we have calves also born from those pregnancies. The problem only remains was that the owners, they don't want the calves to be born in summer months. That's why they say, oh, no, they are seasonal breeders. But I think the females are not seasonal breeders in camels. Maybe the males, yes, because of the heat stress. So if we try to give them a little bit controlled weather conditions, they breed quite throughout the year. This is another big problem, which is brucellosis. So how to use those animals? Because in Dubai, we have some very elite animals belonging to our bosses, which are costing millions and millions of dollars. They don't want to keep these animals unproductive. So we are doing this now routinely, doing embryo flushing from such donors, mating them with the brucella positive bulls also we have, and we get the embryos. So we try to salvage the germplasm also from these donors. In vitro embryo production, that was the in vivo, and the in vitro embryo production is where we produce the embryos from the oocytes and the spermatozoa, or what's the technique we use in the lab, and we transfer those embryos into the synchronized recipients for taking them to term. For in vitro embryo production, we need two raw materials. That is, the first important raw material is the oocytes. So we can collect the oocytes from the ovaries. This is an ovary collected from the slaughterhouse, and you can see the follicle. So you have to aspirate these follicles or rupture these follicles to get the oocytes. So these oocytes are the immature oocytes collected from these follicles, and they need to be cultured for 24 to 30 hours in the lab to mature them to metaphase second stage. This is what we usually. This is their nuclear status when we collect them at the slaughterhouse stage. So this is called a germinal vesicle stage. Then after 8 to 10 hours, the chromatin starts condensing, and they form metaphase 1 stage. And within about 20 hours, you could see most of them in anaphase, telophase, and anaphase stage. And 24 hours, most of them have metaphase second stage, where the chromatin, the oocyte chromatin, and the polar body is separated from each other. This is a typical metaphase second stage oocyte, which you see after 24 to 30 hours of in vitro maturation. We can keep these ovaries. If we collect these ovaries, we have seen that we can keep them for 4 to 5 hours in normal saline from slaughterhouse, but we can keep them for 12 hours at room temperature without any effect on their in vitro maturation. Even we have used these ovaries after now 12 hours, and we can collect the oocytes. If we keep these ovaries at room temperature in normal saline solution, we can collect the oocytes, keep them in pre-maturation media, and store them for 24 hours as well. Because we don't have any access now to slaughterhouses in Dubai, so we are collecting the oocytes from the ultrasound guided transvaginal ovum pickup. So this is the protocol we use for same protocol which we use for the donors for embryo collection. We use the same protocol for superstimulation of these donors, the oocyte donors. So we check them when they have a dominant follicle, they get GnRH, and after the ovulation, which is usually 24 hours after that, after 4 days after that, we start them on the treatment. But now, instead, we are using this one also, but simultaneously, we are using the one which I was talking before, where we use hyaluronan, or we use this as a conjugate to reduce the cost of the hormones. And once the follicles have reached about 1.3 to 1.8 centimeters in diameter, we give them GnRH. Because camels are induced ovulators, so this helps us to collect the in vivo matured oocytes after 26 to 28 hours. So we skip the one process of in vitro maturation by this technique. This is how the transducer is introduced into this transvaginal into the vagina with a needle guide. you hold the ovary with one hand against the transducer, and what you can see is, you can see the follicle, something like that, and you have a needle guide as well. So the follicles, this is a needle guide, you can see the follicles, and aspirating these follicles, this I took from somewhere, maybe from some human fertility center, so to just demonstrate. So this is the needle goes into the follicle, you aspirate this follicle, go to another one, and you keep collecting these oocytes, which are all of them, most of them, about 80 to 90% are matured in vivo. So this is what I was talking about. So if we see that 24 to 25 hours, we have most of the oocytes are about 40% are matured, whereas 28 to 29 hours, about 91% of these oocytes are matured. And we try to see, is there any difference in the development of these oocytes, which we collect from these in vivo animals and which we mature in vitro from the slaughterhouse ovaries. We could see that even the cleavage is same, but the difference is definitely there in the blastocyst formation from the total number of the oocytes. But all those ones which cleaved, whether they are from in vivo matured ones or in vitro matured ones, they form blastocysts at a same rate. So these oocytes, how we produce the embryos, number one technique, in vitro fertilization. In vitro fertilization, what happens is that you incubate the spermatozoa with these in vitro or in vivo matured oocytes. And when the spermatozoa enters the oocyte, it forms something like this. This is an oocyte chromatin. This is a spermatozoa chromatin which condenses, and it forms two pronuclei which they fuse together to form a zygote. This is a typical oocyte where you can see two spermatozoa have entered into the oocyte. And this is typical embryos which we get, a 2-cell embryo, usually 24 hours. In camelids, usually we have seen from 16 to 18 hours, we get these type of embryos. And then morula, early blastocysts, and they start hatching like this, and they are hatched within 5 to 6 days of culture. First dromedary, IVF calf was produced in 2005. 2005, this was the lab of Abdul Haq Anansi from Abu Dhabi, and this is the only lab which produced these calves. We also did some work on using the epidermal spermatozoa. We use these, store these epidermal spermatozoa for up to 8 days in tristless and lactose buffers, and we could produce embryos using this technique up to with no effect at all, even up to day 8 of the storage. But we never transferred these embryos because there was no demand for such type of the offspring production. Intracytoplasmic sperm injection is the technique which we can use in bulls, which are like which we cannot use for natural breeding. Or sometimes we come with the recipients or some elite camels which have some problems and they are not able to breed normally, like they have adhesions or their fallopian tubes, they are blocked. So in those cases, we collect the oocytes by ultrasound guided ovum pickup. We take the spermatozoa from the bull, whichever bull we want, and we do ICSI. And this is a typical, like we take one spermatozoa, you need to inject it. This is a mature oocyte. You can see the polar body. You need to inject this one into the oocyte, and after injection, these oocytes are we call them the presumptive zygotes now because they are injected with spermatozoa. They need to be activated. In camels, we have seen that all these ones which are being injected, they need artificial activation. They don't develop without artificial activation. They develop, but in a very small quantity. We have produced this is a typical how you get from one cell from one cell is in morula an early blastocyst, expanded blastocyst, and a hatchet blastocyst when you stain these embryos with the hex stain. So this is the current status of in vitro embryo production by IVF or ICSI in camels. In camelids, rather. If you see this lab, what I was talking about, they use fresh ejaculated spermatozoa, and they have produced 3 or 4 publications. So they produce only this much, these live animals, and I will leave this one now. So I'll go to the somatic cell nuclear transfer, which is a technique, other technique used for the production of the in vitro embryos. So what is difference between the somatic cell nuclear transfer or cloning and the natural breeding is that in sexual reproduction, the baby or the calf which is produced has half of the chromatin coming from the male and half of the chromatin coming from the female, and the offspring or the embryo which we get, it has half the chromosomes from each parent. Whereas in somatic cell nuclear transfer, we take these oocytes, we remove their DNA and or the chromatin, we take the chromatin from the somatic cells, we inject that into it, and what we get, the embryo is having all the chromatin or the DNA from this somatic cell. So the babies produced are the exact genetic copies of the donor cell. How is it done? So for example, I have taken this picture is in exact, this is a copy of this is a winner of a beauty contest, which was in our center, and we took the cells from this one. We get the oocytes from the ordinary females by ultrasound guided ovum pickup. We mature them, and after maturation, we remove their nucleus and inject this cell in the perivital space. We need to fuse this cell with the enucleated oocyte. Once they are fused, we need to do a chemical activation of these cells, of these presumptive zygotes, or we call them reconstructed embryos. Because this cell normally is programmed to form the, for example, here, this is a second fibroblast cell. So this cell is programmed to form the second cell. So we need to activate this cell. Come on, you are no more. And second cell, you need to start forming an embryo. So you have to become an embryonic cell. So after chemical activation, if we are lucky, we get the embryos, and within 7 days, 6 to 7 days, we get the blastocysts, which are transferred again into synchronized recipients. And if we are lucky to get a pregnancy, we get a baby. And this is the exact genetic copy of same donor. So this is the setup we use in our lab for micromanipulation. First cloned embryo transfer, first cloned baby was produced in 2009, and this is INJAS, which was produced in 2009. After that, in this one, we had used cells from cumulus cells. We have compared cumulus cells. We have compared second fibroblast cells. We have compared fetal fibroblast cells. What we could see is that cumulus cells were giving better results as compared to second fibroblast cells. We couldn't get any pregnancy with the fetal fibroblasts that year. And we got 2 babies produced. One was INJAS in 2009, and other was Bin Sogan, same year in 2010 from the same experiment. So this was the INJAS, world's first cloned camel, and Bin Sogan was the second one from the copy of a elite bull we had at our central camel reproduction center. In the process, what we have seen is that we can use actively growing cells, we can use confluent cells, we can use serum starved cells. Nothing, there is not much difference, but better is to use always serum starved cells than actively growing cells. We use different types of cells, second fibroblast and cumulus cells. This is what I was saying. The cumulus cells give us better results. But now we are routinely using second fibroblast cells because it's so easy to collect the second fibroblast cells than the cumulus cells, because cumulus cells you can get only from females. And if it is an elite animal, they don't allow you to insert the probe into the vagina and get the cumulus cells or aspirate it. So better is to take the skin sample somewhere where the owner even even does not want the other people to know that you have taken a skin sample from this animal. Skin fibroblasts even now are giving us better results because the technique has been improved a lot. We have used the cytoplast, means oocytes from in vivo and in vitro for cloning. And even though in vivo gives us better results, but still we had been able to produce calves, whether it is from the sources, from the live animals, or it is from the dead animals from the slaughterhouse. Both of them are equally good. Only the thing is the in vitro maturation process, it needs a little bit of more work to be done and more optimization in camels. So this another group which used another method, they called it handmade, but I called it a zona-free method of cloning, where they remove the zona, and instead of using the conventional method of the injection, they use a needle which is not having a spike. And they also produced the calves, but they could see that their method, this method was a little bit better in one way, but the conventional method is better in the other way. Because I have been given notes of 5 minutes, I'm trying to rush through the slides. So in the process, you can see that the cloned dairy camels have been cloned, the beauty camels have been cloned, and the racing camels have been cloned. I will not go through it. I'll go quickly through this. And this is the interspecies somatic cell nuclear transfer, where we have used the oocytes. So many people had been working on this initially, like the Chinese groups. They did cattle oocytes with the bacterial cells, and they were not able to produce any blastocysts, but we were able to produce blastocysts when we used the skin fibroblasts from bacterial camels and the in vivo matured oocytes from the dromedary camels or urban camels. And same way we did with the llamas also. We got a good number of blastocysts, and we transferred them. We produced the baby from the bacterial camel clone calf from the dromedary. This is a typical presentation where you can see the cells were from this double hump camel. This was used as a nucleus, the nucleus would be for this, oocyte donor, and the baby was produced from the dromedary one. This is the publication. This is the actual baby, but you can see the condition of the mother was very bad. So it happened during our off season when we had nothing to do, and we did this type of the work because there was no demand from our bosses for this type of the work. So in the process, so this is what I wanted to emphasize, that in the process we have cloned the racing champions, which, like the prize money for a winning camel is upwards of $2 million. We have cloned the beauty, they call them beauty queens, or the winners of the beauty contests. The prize money for this Al Dhafra festival last year was more than $66 million. And the breeding, we have cloned breeding bulls. Breeding bulls cost about $10 million. In Dubai, we have cloned many these milking camels, which is a multi-million dollar industry. We have cloned the bacterial camels, and we have cloned some of the camels which have died 3-4 years, or died few days before we were asked to take samples from them. Even I remember one case where I was being asked 3 days after the death, and the animal was even buried under the sand. They took the animal out from the sand, sent us an ear from cut an ear from the animal and sent us that ear, and we were lucky to get some live cells from that, and we gave three babies to the owner for that animal. So we called it the resurrection of the dead camels as well. This, I will take just 1 minute. This was like yesterday, some questions coming, and someone delegate was asking about this. So that's why I wanted to add this, that biotechnology has always been accepted. There have always been challenges before it was accepted in the mainstream reproduction. So this is like when the pasteurization of the milk was introduced in 1992, you see, this is the quote from the Introduction of Pasteurized Milk Ordinance, which was, I mean, the report. What they said is, it's unknown what long-term health consequences may unfold. The studies are not adequate. Furthermore, this will not be available and cost-effective for small farmers, and it will decrease product acceptance and consumption. And nowadays, I don't think any of us will be drinking raw milk. We always will love to have the pasteurized milk. Similarly, this one, when the first baby in 1978 was born, the Louis Brown from IVF. And what the people were saying, we are talking about the possibility of human-animal hybrids, monstrous babies, children born this way will have no soul. So these are the things which were, before accepting a new technique in the mainstream, these type of things will come up. But we have, as a scientist, we have to come through these hurdles, and we have to show that, no, this technique is really useful for us, and these techniques can be used for enhancing the production and multiplication of our elite breeds and improve the economics of our livestock farmers. Conclusions, I will go quickly through them. So we should use these breeding techniques for enhancing meat and milk production in local breeds. This is local breeds. So embryo collection and transfer is now well-established technique, and I will suggest that the granting agencies in Morocco, they should finance the projects on embryo collection and transfer for enhancing the genetic gain in camels of the local breeds. In vitro embryo production has been developed, and the application of cloning to camelids has opened doors for multiplication of these elite animals, and we can use it also for the modification of the desired modification of the genetic modifications and related technologies. Cell banks should be established everywhere to have these, to keep the diversity of the genetic pool. And embryo freezing or encouraging. Recent studies from the Lalou's lab are very encouraging, and they could be used in the future. And use of desired genetic studies and modifications which could help in obtaining the desired objectives for milk and meat production, and to improve the profitability of the camel husbandry should be emphasized on. I will thank you for your patience, and I'm happy to take any questions.
Thank you very much,
Sorry for
Dr.
being,
Weni, for this
2 minutes
insightful
late.
presentation about the cutting-edge production technologies. I just open the stage for one question, and maybe people can discuss with you later, because we need to move on. So please, if you have any question, yes? I know that we will have a lot of questions, but yeah.
Thank you so much. It's a wonderful presentation. If you can hear me well, right?
Thank you, sir.
Yeah, thank you. So I share the concern of the embryo transfer and all of this using of the somatic cells with the people, because like the law is not reaching to that level to modulate and monitor what we are putting in the bank and what we are using. You have done a great job. I am impressed with the number of publications and the way you addressed all the aspects of the embryo transfer and the fertilization. You mentioned something about the sperm chemical activation, which I call it capacitation, because it's decapacitated in the epididymis. So can you elaborate a little bit more about the chemicals you used to activate or capacitate the sperm? Thank you so much.
Thank you very much. In this presentation, I think what I was talking about, the chemical activation of the reconstructed embryos in cloning process. Yes, we do need the chemical activation for spermatozoa. I mean, we call it capacitation of the spermatozoa for in vitro fertilization, or whether we do ICSI. And we use heparin and calcium ionophore in camels. But here I didn't mention that because I knew I have to rush through these presentations. But that chemical activation is because we are using a sickened fibroblast cell, which is programmed to form chicken. And we are using the nucleus of chicken fibroblast into the enucleated oocyte. And we need to do chemical activate the chromatin of this somatic cell to, that's what I was saying, somebody was saying it like, this cell is sleeping and you need to just give her like a stimulus, something, come on, wake up, you are no more a skin cell. You have to start forming an embryonic cell. So that's what we use, calcium ionophore or ethanol, or sometimes in some species, electric stimulus for activating the nucleus in these reconstructed embryos. Thank you.
Thank you very much, Dr. Wani, for this
Thank you
insightful
very much,
presentation.
everyone.
People reach to, all the people that want to ask questions, reach to Dr. Wani. He's here for that. Thank you very much. Please applaud Dr. Wani again. And now, welcome with me Dr. Skidomer Julian, which is Scientific Director of Camel Production Center in Dubai. Please, she will tell us about the advances in artificial insemination in camels. Please, you have the stage. Thank you. You can use that or that as you want.
I'll use this one.
Or we can both. if you want.
Both. Thank you. Where's the pointer? Have the pointer. Yeah. Thank you. Need that one. Good morning, ladies and gentlemen. Hello. Right. Good morning, ladies and gentlemen. Thank you, Mr. Chairman. And it's a pleasure to be here today to share with you some of the work we've done with artificial insemination in camels. So first off, the first question most people ask is, why do we need to do artificial insemination in camels? And can't they mate naturally? Well, of course, they can mate naturally, but there are many advantages to doing artificial insemination. First of all, it'll give you more efficient use of your genetically superior male. You can collect the semen, extend it, and then inseminate 2 or 3 camels rather than it just mating 1 camel. It helps to prevent diseases, can eliminate the need for transportation of animals, because obviously a pot of semen is much more easy to transport than an animal itself. It can eliminate behavioral problems. We do have male camels that take a particular dislike to certain females, and they won't go anywhere near them. Sometimes they have leg problems or joint problems, and they can't crouch behind the female and mate. So again, artificial insemination would help these problems. And also, if we can get to the point where we can preserve semen, you'll be able to extend the reproductive life of the good males, because it can even be used after the death of the male. However, AI isn't always that simple. There are certain current limitations to using artificial insemination. First of all, camels are seasonal breeders. They've got a short breeding season in Dubai from October to the end of March. You have to train the male to be able to collect semen, and that's not always that easy. They don't always like the artificial vagina. And if we can train them to the artificial vagina, we then use them just for artificial insemination and don't let them go back to mating naturally. Once you get the semen, it has a low volume and a low concentration, so it's difficult to work with, and the viscous nature of semen makes it even more difficult to work with and to get proper motility results. Obviously, the viscosity makes mixing it with extender difficult, and even now we have a lack of standard techniques for freezing semen. And it's not only the male you have to worry about, you have to worry about the female as well. You have to be able to induce ovulation in the female before you inseminate them. Now, the tried and tested method of semen collection that we use is the artificial vagina. It's a modified bull artificial vagina. You can see there's a bit of a cervix that we put at the end of the AV. The semen gets collected into this test tube here, which is water jacketed, because the semen collection process can take 15 or 20 minutes. So you have to keep the semen warm during that time. We use a estrus female or receptive female and let the male mate her naturally or get on behind her, and then we just guide him into the artificial vagina. Okay, okay, I'll slow down, so I hope we try to get through these slides. Okay, so the artificial vagina is the way that we tend to use. However, other people have been using the phantom, trying to develop a phantom. Not always do you have a receptive female. And a friend of ours, Dr. Amir Nizari Nazlaji in Iran, has developed this phantom here that you can see. Doesn't look too bad. It looks pretty much like a camel. It's covered in camel skin, and then the artificial vagina is inside here. However, when our camels got on it, it's made of fiberglass. So when our camels got on it, it made a bit of a clunking noise, and they didn't like it very much. But he did have a female male that would use it, and he could collect semen quite regularly. Another company came to us with a different type of dummy. This one has a spring underneath it. So it even had a neck that would move. They put a tape recording underneath here of a camel gurgling, thinking it would be more effective for the male. However, our males took one look at this wild eye of this camel and ran in the opposite direction. So this dummy obviously needs a bit of work on it. And more recently, they've been trying to use the camel condom. And Dr. Anouassi and his group, and in Saudi Arabia, they're now using it on quite a regular basis. They have a sleeve, which they use this metal probe here they put inside the sleeve and then insert that into the vagina of the camel. And then you have to attach the outside of the condom to the camel. Anouassi's group use a harness, which I think is probably a bit better. We've used tape, but that does tend to come off, especially when it gets wet. We haven't had much luck with the condom, but I do think it's a method of semen collection that is worth progressing with, because other groups have had more success, and I think some males that don't accept the artificial vagina might well accept the condom, but they do have to be trained for this too. Once you have the semen, you have to be able to assess it, and we look at things like viscosity, motility and concentration, and morphology. For the viscosity, we measure it by either grading it 1 to 5, by saying how difficult it is to pipette up and down. So if it's got no viscosity and it's easy to pipette, it's graded at 1. If it's got very difficult to pipette, then we grade it at 5. Or you can grade viscosity by drawing the semen up in a pipette until breaking point. So you measure it against a ruler, and once at breaking point, that is the viscosity measurement. Then we look at motility and concentration, and for that we use a CASA, which is this fancy machine here, or hemocytometer. And then if you want to do morphology, we do the simple irisin nigrosin stain, where the damaged sperm then stain dark pink. When we're assessing sperm, we also look at membrane integrity. And for this, we use a hypoosmotic test, where the semen is diluted with a hypoosmotic solution, incubated for 45 minutes. And then or we can use propionate iodide. With the osmotic test, you can see here the sperm intact membranes are coiled and the non-intact membranes are straight. And if you're using propionate iodide, then the intact membranes stain green and the non-intact membranes stain red. You also have to look at acrosome membrane integrity. The acrosome is like the cap on top of the sperm head, and you have to look at the integrity of that when you're assessing the sperm. So again, we make smears of the neat or diluted semen, stain it with FITC-PNA, and then you can see that the non-capacitated sperm have green acrosomes, the capacitated sperm have a green equatorial zone here, and the damaged chromosomes don't stain at all. It's not only what the sperm looks like, it's whether it is actually functional too. You've got to be able to fertilize. So to measure the sperm function, we do a sperm penetration assay. Now, ideally, you'd want to use camel oocytes, but there's no real slaughterhouse in Dubai that regularly slaughters the camels. So we use goat ovaries instead, because we can get goat oocytes much more quickly. Once we've retrieved the oocytes, they're matured overnight, and then they're denuded the next day to remove the cumulus and the zona pellucida. The denuded oocytes are then incubated with sperm for about 18 to 24 hours, and then they're fixed with lacmoid, so that we can measure the number of sperm bound, penetration rates, and the formation of the pronuclei. And when we've used this in our lab, we've found that 79% of the sperm will fuse with the oolemma, and 67% of sperm successfully penetrated, decondensed, and formed pronuclei. So these are the goat oocytes prior to maturation. This is after 24 hours of maturation where the cumulus is expanded. And here you've got the penetrated head with a detached tail, and the decondensed head. So once you've looked at the semen and assessed it, you obviously have to process it before you can inseminate. And as we've been hearing earlier as well in the earlier lectures, the viscosity of the semen is one of the major problems with AI in camels. Semen doesn't spontaneously liquefy, and unlike stallions and pigs, there's no gel fraction. You can't tip off the gel and then have a sperm-rich fraction left. So the sperm are actually entrapped within this gelatinous seminal plasma, which makes the motility assessment really difficult. So how do we liquefy the sperm? Well, you can leave it on the lab bench for 20 to 30 minutes, but we don't find this really helps. You can put it in an incubator for 20 to 30 minutes, but you have to watch out that incubating sperm, they don't burn out. So you can use mechanical methods, like pipetting and vortexing or centrifugation, and we found that
gentle pipetting is usually the best method, does the least damage to the sperm. Or you can use enzymatic methods, use all these different enzymes. And again, we've found that papain is the best enzyme to use. So David Monaco and his group in Saudi have looked at using papain to reduce the viscosity, where they added papain to the diluted semen, pipetted up and down to reduce some of the viscosity and incubated it for 5 minutes. You then have to add a papain inhibitor to stop the reaction before you assess the sperm for the motility and agglutination. And they found that the papain-treated samples showed a significant increase in progressively motile sperm, but they also showed a slight increase in agglutination. And agglutination is when the sperm head sticks together. So that's not good if you want to inseminate the sperm. So once we need to process the semen for AI, this is just to summarize what we would do. You dilute the semen, first of all, 1:1 or 1:3, depending on the concentration and volume you had to start with. Repeatedly pipette it up and down to reduce the viscosity. And then at this point, you can evaluate motility and concentration and inseminate if you want to. However, if you want to further improve the sample, then you'd have to centrifuge it, resuspend it, the pellet in green buffer, before assessing it again and inseminating. Normally, we inseminate between 150 to 300 million live sperm. And it's interesting to note that in actual fact, even though the centrifugation will remove the seminal plasma, it doesn't seem to affect the AI results. Whether it's centrifuged or not, we get pregnancy rates of around 50%. However, centrifugation does cause damage to sperm. So we've looked at methods to try and reduce this damage. And the horse people in the equine world use this cushion. So we've had a go at trying to see whether the cushion would affect the motility and vitality of camel sperm. So the method we used is we put 60 mils of cushion into the bottom of a tube, put your diluted semen on top, centrifuge at 60 G for 10 minutes. Then you remove the supernatant, and we resuspend the pellet. We actually resuspend the pellet with the cushion. We've got such a tiny amount of cushion there that we don't bother trying to suck the cushion off. And then you can find that with our cushion centrifuge samples, we got improved, all parameters were improved, the total motility, progressive motility, vitality, and acrosome integrity. What we did find as well, though, when we looked at the pregnancy rates, there was no difference in the pregnancy rates between the cushion and non-cushion sperm. However, the numbers are so small that it's impossible to actually get any significant difference there. But what it does show is that the cushion didn't have any deleterious effects on the semen and the fertility of the semen. David Monaco, again, has tried cushion as well. He used a slightly different technique where he used a larger volume of cushion. He put 1 mil of cushion in the bottom of the tube, layered on top his 8 mils of semen, centrifuged at a higher speed for a longer time, 900 G for 20 minutes, removed the supernatant, leaving just a bit above the sperm buffer layer, which is here, and then aspirated the cushion. So he removed the cushion before he resuspended the sperm buffer layer. And in his results, as you can see, he also, with the cushion centrifuge samples, got an improved progressive motility, but his other parameters were much the same, whether or not they had cushion before they centrifuged it. However, he continued that study using papain as well prior to centrifugation, which in other words would make the semen more liquid before they centrifuged it. And you can see this time he did get better total motility and progressive motility. So papain-treated semen definitely showed increased motility parameters. If you want to then select your viable sperm, you have to go one step further. And obviously, if you're using sperm for IVF or you want to freeze it, then you need viable sperm to start with. So we've done this study in conjunction with Jane Morell and Uppsala University in Sweden, where she's developed various colloids for lots of different species. She's developed this colloid especially for camels. So what we did is we dilute the semen 1:5 or 1:10 in your extender and try and mechanically liquefy it by gentle pipetting to release the sperm as much as possible. We then layer the semen over 2 mils of colloid, centrifuge at 300 G for 20 minutes, and then after centrifugation, you discard the supernatant and colloid before resuspending the pellet. And you can see here that in the SLC, or the single layer centrifuge sperm, we got an increase in the total motility and progressive motility, whatever the dilution was, and also an increase in the membrane intact sperm. So we can tell you that SLC, single layer centrifugation, certainly seems to select the viable sperm. However, the disadvantage was that the sperm recovery is quite low. And this is because you're losing sperm, obviously, within the supernatant, and you're losing some sperm through the colloid. So the colloid certainly is useful, but I think we need to do further investigations, probably looking at different colloid formulations, the size of the tube, centrifugation speeds, colloid and semen ratios, all those to see if we can improve sperm recovery. So once you've got your sperm, you've assessed it, you've handled it, then you need to inseminate it. And as we've mentioned before, the female camels are induced ovulators, so you have to inject GnRH or HCG at the right time of the cycle, which means they have to have a mature follicle measuring something between 1.3 to 1.7 centimeters in their ovaries. It's no good inseminating camel at any point of their cycle. They have to have a follicle here that's ready for ovulation. So we inject GnRH, HCG to make them ovulate, and then inseminate up to 150 or maybe up to 300 million live sperm, either at the same time or 24 hours after the GnRH injection. And we get marginally better results when we inseminate the semen 24 hours after GnRH injection, of around 50%. And that's because we know when we've scanned camels every 2 hours, that they tend to take between 26 and 32 hours to ovulate. So obviously inseminating them at 24 hours after GnRH is going to be nearer the time of ovulation. However, if you don't want to inseminate the semen straight away, you might want to send it to a different farm. Maybe your female isn't ready for inseminating that day, and maybe it's going to take another 12 to 24 hours for that follicle to mature. In that case, you want to be able to preserve your semen. And there's 2 ways of preserving it, by either cooling it 4 to 5 degrees, and that will only last for up to 24 to 48 hours, or for freezing. So for short-term semen preservation, we slow cool it to 4 to 5 degrees, and you can do that by either placing it in a refrigerator in a beaker of water, because that'll slow the actual cooling rate. You can't just put it straight into the fridge, because obviously the cooling rate would be too aggressive. Or we can use this, which is an equine equitainer, which gives you a much more regularized cooling rate. You've got 2 cooling bricks here, which go at the bottom. You put your tube of semen inside this plastic cup, which goes inside this polystyrene sleeve, and you put it inside your equitainer. And the results, as you can see here, from Maidan's group and Clara Mallow that was working with us, we've got results of between 22% and 66%. But here, even though our results were quite promising with Clara's working with us, 4 out of 6, the numbers aren't great. So I wouldn't say that it was always going to be that good. So what can we do to try and improve it? There's always room for improvement on those success rates. So we looked at catalase. We know that catalase is an antioxidant, and we know that when sperm is being stored, it's undergoing anaerobic metabolism and producing ROS, or reactive oxygen species, and toxic byproducts like hydrogen peroxide. So this group, Maidan et al., back in 2008, they looked at various concentrations of catalase, and they came up with 500 units was the best concentration to use, and they stored the semen from up to 5 days at 5 degrees centigrade. And you can see here that even at 5 degrees centigrade, at 5 days of chilled sperm, they actually still maintain motility. So by adding catalase, they were able to improve the motility of stored sperm, and they got to reduce the number of dead sperm and abnormal sperm. So they came to the conclusion that catalase can improve camel sperm survival during storage at 5 degrees centigrade. And another group, Sayed Asghari, back in more recently in 2024, looked at the effect of another antioxidant, N-acetylcysteine, or NAC, on the effect of liquefaction and fertility of chilled sperm. In the first such experiment, they diluted the semen in an extender containing 0, 6, 12, or 24 millimolar of NAC, and then assessed the viscosity at 5, 15, 30, or 120 minutes, up to 24 hours. And they found there was a significant drop in viscosity at all time points, with a dose-dependent effect. In the second experiment, they diluted semen in extender containing 0, 6, or 12 millimolar of NAC, and then looked at the viability, motility, and DNA integrity at all these time intervals up to 48 hours of chilled storage at 5 degrees centigrade. And here they found that the semen extended in 6 millimolar of NAC showed better DNA integrity at 2 hours and better motility at 48 hours. But concentrations above 6 millimolar lowered the viability and motility results. So to do the final test is obviously to see whether or not you get any pregnancies. So they then added NAC to semen extender and inseminated 60 camels with chilled semen that had been chilled for 26 to 48 hours. And these are the results. Sadly, it didn't have any great effect. There was no real difference between the controls and NAC. But what it does show is that NAC did not affect the sperm freezing fertilizing ability. So NAC was okay to use, and it does seem to improve the liquefaction and other factors. So they came to the conclusion that the mucolytic property of NAC is beneficial for partial liquefaction of the semen. Adding NAC to the semen extender certainly seemed to improve sperm quality and functionality during chilled storage. The optimum concentration of NAC was 6 millimolar, and higher concentrations of that compromised DNA and functional integrity. So if you want to preserve the semen even longer, then you would have to cryopreserve it. Cryopreservation, with many stages. You have to collect it, cool it, add your cryoprotectant, freeze it, thaw it, and then inseminate it. So first off, you collect the semen, as we've described already, and then you would dilute the sample 1 to 5 in your first off semen extender. Then we try and liquefy it by mechanically, by pipetting it up and down in a pipette. But we always get low motility. Camel semen is known for its low motility, so we would run it through the single layer centrifugation process, where we run it through the colloid, again, centrifuging it at 300 G for 20 minutes to select those viable sperm. Once you've got your pellet, then you have to do a 2-step dilution factor. And the extender we tend to use, that we always find as our gold standard, is green buffer, and we add 20% egg yolk to that. So when we've got the pellet, we resuspend it in your fraction A, we call it the green buffer and 20%, and then you cool it to 5 degrees centigrade over a period of 2 hours. You then have to look at what cryoprotectant to use. And we've tried various cryoprotectants like glycerol, ethylene glycol, methylformamide, and DMSO. And we found that glycerol and ethylene glycol gave us a better motility rates. This is work we did with Clara, who was working with us for about 7 years. However, another group have looked at it as well, and they found methylformamide. Al-Badri's group found methylformamide gave them better results. And obviously, it's not just the cryoprotectant that you have to use, it's the concentration of that cryoprotectant. So again, we've tried different concentrations, 1.5, 3, 6, and 9% of the cryoprotectant. We found that 3 and 6% gave us the better results. So once you've got the cryoprotectant and the concentration of the cryoprotectant, you then think, well, how long are we going to let it equilibrate? We've tried various times, 5 minutes, 30, 1 hour, and 2. And we found that 30 minutes gave us the better results. So obviously, in summary then, we use glycerol at 3% for 30 minutes equilibration. And during that equilibration time, you have to load it up into your semen straws whilst it's still at 5 degrees centigrade before you freeze it. And the freezing rate is the next thing to consider. And we float these racks over liquid nitrogen. And obviously, the height of the rack will affect the actual cooling rate. So we've used 1 centimeter, 5 centimeters, or 7 centimeters height above the liquid nitrogen, or you start at 5 and then reduce it to 1, or you can start at 7 and reduce it to 5. And we've found that the 1 centimeter above liquid nitrogen gave us the better rates, which means that obviously it's the faster cooling rate. So once you've frozen it, you plunge it into liquid nitrogen, and then you can store it in your tank for as long as you like, until the females are ready, or until you want to use it. Obviously, before you inseminate it, you have to thaw it out, and there's been 2 methods of thawing, either the 37 degrees centigrade for 30 seconds or 60 degrees centigrade for 10 seconds. And again, we found that actual fact, the higher temperature for the shorter period of time gave us better results. So just to summarize our semen cryopreservation method then, we dilute the semen 1 to 5, run it through a single layer centrifugation to select the good sperm, do a 2-step dilution, diluting it in green buffer and 20% egg yolk, cooling it to 5 degrees centigrade over 2 hours. We then add our fraction B, which is the same extender but with the cryoprotectant, and we use 6% glycerol at a 1-to-1 ratio, which gives you a final concentration of 3% glycerol. We would then equilibrate that for 30 minutes. During that time, you load the straws in 5 degrees centigrade, freeze at 1 centimeter above liquid nitrogen, and then store it in your tank. And then when you want to thaw it, you will thaw at 60 degrees centigrade for 10 seconds. However, even now, and after many years of persevering with camel semen, pregnancy rates are still low, and they're still less than 10%, in whichever group seems to have done it. Nobody's really successfully found a commercially available method of freezing semen. So then in summary, I would just like to say, semen can be collected with an AV, but also a condom is now showing promise. Viscosity is best reduced mechanically or with papain. Centrifugation with cushion improves all parameters. Running it through SLC, single leg, the centrifugation through a colloid selects viable sperm. Using a sperm penetration assay is using heterologous oocytes like goat oocytes can be a measure of sperm function. Using NAC or catalase antioxidants added to chilled sperm improves motility and quality. And whilst it is possible to obtain pregnancies with chilled semen, acceptable pregnancy rates and fertility of frozen camel still remains a challenge. So, and I would just like to finish with saying that we've been talking a lot about the fancy techniques we can use, and with Vani's lecture before as well, with the AI, the embryo transfer, the cloning. And we mustn't lose sight of the fact that we want these methods that we can use in the lab to be able to be used by those in the field. So don't be put off if you can't use artificial insemination yet, or embryo transfer and anything else. You can certainly improve the production of your females just by monitoring the cycle and mating them at the right time. And as I think I mentioned on yesterday in the debate, it's important to first off start with a healthy herd, get your herd healthy and fit and well fed, and then look at just monitoring the cycle, making sure you're mating those females at the correct time, when they've got a follicle in their ovaries. It's no good just mating her when you think she's receptive, because camels' estrous behavior doesn't relate to follicular growth. They will sit and get mated at any point in time, even when they're pregnant sometimes. So monitor the camels. If you can, use ultrasound. That's the tried and tested gold standard. But if you haven't got that even, then just mate your camels twice, 24 hours apart, every 15 days. They will ovulate at some point during that cycle. If you don't know they've got a follicle the first time you mate them again 15 days later, the chances are they'll have a follicle. And if they haven't then, mate them again 15 days later, and the chances are by the third time you'll strike lucky and you should get them pregnant. And it's not a question of trying to mate them in between. Be strict, just do it every 15 days. If you mate them when they have got a CL, you run the risk of getting them infected. So they've tried this study, we've tried it with camelicious people as well, and it's a 15-day fixed time period. Doing it at 15 days, you should get your camel pregnant. And once you've got those pregnants, then you can start on the fancy things like artificial insemination and embryo transfer. And so good luck with your projects. We're here to help, and we'd be happy to help in any way we can. And I would just like to say, just before we finish, I'd just like to say, obviously all this work is a team effort. I'd like to thank all the boys that handle the camels. This is Clara, who's done a lot of work with us on the semen over the last 7 years. Brendan came when Clara left and continued the work. This is the lab team. And of course, these are our beloved camels, which we couldn't do without. Thanks very much.
Thank you very much, Dr. Skidamore, for this very insightful presentation. So we have time, because she respected exactly the time. So we have time to ask some questions. So please, if you have any questions, go ahead.
Thank you, Lulu, for this nice presentation and the recommendations. I have two questions. The first one, as you know, that fertility is not usually linked to the motility, for example, we can find that a bull can be have a good motility, progressive motility, but when it comes to the pregnancy rate, it has no correlation. This is my first question. My second question is about the seminal plasma. In small ruminants, many times we use seminal plasma, mainly proteins in the seminal plasma, to get more fertile males, to detect if this, because there is some proteins that are really well linked to the fertility. Have you tried this method or not? Because I looked for some papers on proteomics, for example, about seminal plasma in camels, but I didn't succeed to get some. Thank you very much.
Thank you for those. Thank you for those questions. On the first question, which was the fertility, I think you must also just look at the fact of the female. It's a two-way situation. It's not just the male. It's nice to blame the male, but in actual fact, the female has a responsibility too. And I find a lot of times people are just inseminating camels or animals without really knowing what stage of the cycle the female's at. So before you blame the male entirely, look at the female. If he's actually mating, if he's running loose with the herd and choosing his own time to mate them, and obviously he knows best, and he's still not getting them pregnant, then you'd have to start looking at genetic problems, fertility, maybe infection problems, and all that sort of thing. As for seminal plasma, we haven't looked at the protein, so I wouldn't know about adding them back, and I don't know anybody else that has. But I know people have, especially when they centrifuge the sperm and with frozen sperm, have looked at putting seminal plasma back in with the semen sample before they inseminate it. I don't think there's been any mind-blowing results. Some people say it helps, other people say it had no help. So we're a bit still, I think it needs further studies and bigger numbers before we can come to any conclusion.
Thank you, Dr. Skidmore. When we talk about biotechnology, firstly, we think about artificial insemination. The breeder later puts question about artificial insemination. How to apply artificial insemination in the field? Is someone applying artificial insemination in the field? Is there any calf born by frozen semen? Thank you.
Just to go in reverse order, yes, there has been a calf born by frozen semen. We have had 1 or 2 calves, but at the same time, it's almost by the luck of the draw. We think, wow, what did we do that time? What did we do differently? And we can never come with any particular method that we used that was different. So, see, there have been calves born by frozen semen, but again, as I say, there's not a commercially available system to be able to do it, that we can routinely say, do it this way, and you will get, you've got a 50% or better chance of getting pregnancies. As far as artificial insemination in the field, I know it's something in Dubai that they want to start. I mean, at the moment, we're still just doing it in the lab. But one of the big problems in Dubai is overuse of the males, especially the good males. The good racing males, the good milk-producing males. Everyone wants to use them, especially the racing ones. They end up with 10 females to mate a day. And obviously, that's not going to work. By the time they get to even number 2 or 3, they're probably firing blanks, as they say. So AI would be particularly useful in that perspective, because you could click the semen, extend it, and then inseminate 2 or 3 camels rather than having the male mate 10 camels in a day. So it's definitely a place for AI. It definitely needs to be used, and it should be further studied. But at the moment, I think the drawbacks are the actual semen collection process, because before you can inseminate it, you've got to be able to collect it. And we find that with our males, you have to use a young male that hasn't used for mating much to be able to get him trained onto the artificial vagina. When we do get them trained on the artificial vagina, we just use them for that and don't let them go back to natural mating, which a lot of owners wouldn't like, because they like the natural mating technique. So you've got a bit of to get through to the owners about artificial insemination, and then you've got to be able to collect the semen. That's why I think things like this condom might be a good method of using it, because a lot of males that don't like the AV might be able to use the condom, but they still have to get used to that. But some groups are having promising results, so I think it's worth persevering with that one. And then once we can collect the semen regularly, that will be the time that people will be able to take it to the field.
Good. I have just one last question, just small question, because we have 1 minute more. So my question is, what is the chances to get artificial insemination rates in camel near those of other livestock species, for example, 80–90%? Because I have seen, I liked your results, but I didn't see the variance of the results. I mean, how much we can...
Yeah, I think it all depends on the male camel that you're collecting the semen from and the fertility. We get usually they say around 50% pregnancy rate with artificial
Our
insemination.
chance to get 80, 90%. I mean, there is a lot of places to improvement.
Yeah, well, there's always plenty of time to improve it. As I said, it's the assessing the motility, the viscosity, the volume, because the work that makes it difficult is you get low volume, low concentration. With cattle, you get so many billions of sperm. With horses, you get hundreds of mils of sperm. With camels, you know, we're lucky we get between 3 and 8 mils of sperm, low concentration, very viscous. So we've got a lot of challenges with camel sperm, but we're going to persevere. And there's always room for improvement, and I'm sure we will get there one day.
Thank you very much. Please applaud with me, Dr. Skedima. Thank you very much. Yes, you have a question. Yes, please go ahead.
بسم الله الرحمن الرحيم. شكرا للأخت الدكتورة على هذا العرض القيم اللي هو
I will translate. Yes. تفضل.
شكرا للأخت الدكتورة على هذا العرض القيم اللي هو عرض يعطي نتائج وهو يتأقلم مع هاي التكنولوجيا المتطورة. بالنسبة الدكتورة بغينا نسولوك، احنا في الأقاليم الجنوبية عندنا جل الإبل هي في مراعي مطلوسة، وهذه التجربة اللي طبقتوها على أرض الواقع وعطات نتائج مهمة. نسولوك واش أكثر مردودية من هذا اللي نعدلو احنا بالنسبة عندنا الجمل نعلفوه خلال موسم الصيف، وفي شهر أكتوبر نطصوه على النوق، ويعني عندنا احنا نعدلوا حالات بهذا الشيء اللي الطريقة اللي في مرحلة تجاوز. عندنا بعض النوق اللي يقوموا عملية غير استوائية، وهذا كيعطل تجاوزهم في ما يتجاوزوا إلى الصيف، والله كاين بعضهم نوع منهم إلى الصيف. بغينا نشوفوا هاي التجربة واش أحسن هي هاي التجربة اللي هذا التلقيح الاصطناعي ونخفضوا من هذا الشيء اللي في المراعي. وشكرا.
You got it?
Sort of. Hang on. A bit bizarre. Okay. I think you're asking about the season. Is that right? The season of doing the artificial insemination and how we can use that to be productive. Well, camels are seasonal breeders. Our males in particular do not produce semen, do not want to mate or anything during the off-season, during the summer. So to try and do things out of season would be difficult. And as I say, to try and improve the productivity out here, I mean, yes, we've got to develop artificial insemination, but it's got to start with the semen collection process and then the processing and monitoring the females. The important thing is to monitor the females as well, because as I say, it's a two-way match here, and they are to blame as well, or part of the story. So you've got to use the females as well as the males.
help you for this answer. فين هو الأخ اللي يسول؟ أخ، أنا في المحاضرة ديالي لقيت قلت واحد الكلمة، قلت هو التلقيح الاصطناعي هو مجموعة أشياء، هو حزمة ماشي حاجة واحدة، وهذا الشيء اللي أوكي نقرب أخ. Thank you very much. هو معادلة فيها بزاف الحويجات اللي خصنا ننجحوهم. إلا خدمنا عند الجمل باش ناخذوا منه زريعة وسوقها مزيانة، خصنا نخدموا عند الناقة وحتى هي نكونوا عارفينها راه عندها بيضة مزيانة. خصنا نجمعوهم بجوج. اليوم باقي أنا في العرض ديالي قلت راه باقي ما وصلناش لهاد الناس، ما غنقدروش اليوم نطبقوا التلقيح الاصطناعي غدا عندنا. حنا باقيين خصنا نخدموا على بعض الأشياء. اولا خاصها تكون عندنا ضيعه التجارب، وفي هاد ضيعه التجارب يمكن حنا نستعملوا هذا الشيء اللي كتعاود عليه هاد السيده، هاد الاستاذه كتقول بان راه الزريعه خصنا نغسلوها ولا خصنا نزيدوا لها واحد الماده لان هي ثقيله شويه. ماشي بحال انا كنخدم على الزريعات الحولي وديال الغنم منذ سنوات، 20 سنه هذه درت اكثر من 5 دكتورات ولا 6 دكتورات غير في الزريعه ديال الحولي. ماشي نفس المشاكل، ولكن قد ما غادي ندخلوا في التدقيق ديال التلقيح الاصطناعي، نتوما ككسابه اللي خصكم النوتي، خصكم واحد الوسيله تحسنوا بها، هو احسن وسيله تحسن به من الناحيه الجينيه، من ناحيه الانتاج. هي وسيله نقدروا نمشيو بها بعيد، ولكن باقي خصنا نشتغلوا عليها، ياك؟ اذا الاول اللي مطلوب منكم نتوما كسابه هي انكم تنظموا وديروا الضيعات اللي يمكن لنا ندخلوا فيها هاد التقنيات. ما يمكنش انا نمشي ندخل تقنيه من هذا الحجم اللي غتطلب بزاف ديال الوسائل وانا كنجري على النياق وعلى الجمال في الخلا، غير ممكن، ياك؟ اذا الا بغينا اليوم خصنا زعما من هذا المؤتمر تكونوا خرجتوا بواحد النتيجه. شنو هي اننا نقدروا نتكتلوا ونديروا ضيعات نموذجيه. ركزت بزاف في الاخير، حنا واخا غنديروا ضيعات نموذجيه اللي غادي يسهل فيها التتبع، اللي غادي يسهل فيها البحث، ما غنقدروش نقولوا لهذوك الناس الاخرين اللي عنده 20 ناقه 30 ناقه طالقهم في الغير مكتف في الخلا ولا في المرعى في المرتع، لا عافاكم ديروا. لا، هذوك حتى هما غادي يبقاو، حتى هما عندهم المكان ديالهم، ولكن المطلوب منكم هي ان الناس اللي عندها الامكانيات تحاول انها تكتل ودير ضيعات تسهل علينا بها احنا الابحاث كاساتذه، وتسهلوا عليكم نتوما كطرق للانتاج للتحسين. وهذه هي الطريقه الوحيده اللي غادي نمشيو بها بعيد. الا بقينا بهذا الشيء اللي كاين دابا، زعما الصراحه حنا كيصعب علينا الحال اننا نديروا دراسات، وانتم كيصعب عليكم الحال انكم تاخذوا التقنيات.
استاذتي فهمناك وفهمنا اللي قالت الدكتوره. يا غير احنا اللي بغي السؤال كان هدفه واش المردوديه اكثر في هذا اللي يعني الابحاث اللي وصلت لها الدكتوره ووصلوا لها العلماء، والله في ذا اللي واحدين احنا فيها ارتيزانات. هذا هو السؤال لب الموضوع. اما ما يمكن عنك ذا اللي قلتي عنك تمي تروغي الناس في البوادي في الخلوات. احنا ممكن نمشيو معاك في ذا النموذج ديال الضيعه، يا غير احنا اللي سولنا عنه كان اكثر مردوديه ذا اللي وصل له العلم في قضيه تجاوز النوق بالحاله بهذا الاستنساخ ونقل البويضات وهذا النوع من ذا الشيء اللي واحد فيه احنا اصلا ارتيزانال ووارثينه. وشكرا.
الجواب على السؤال ديالك هو انه اللي خصك تعرف بان التلقيح اصطناعي. وانا البارح قلتها لواحد الاستاذ، قلت له الى اليوم جميع المغاربه عندهم الحق يدخلوا لاي سوبر ماركت ويشريوا الحليب باش يدوشوا ماشي باش يشربوا، ياك؟ سكغاس التلقيح اصطناعي، ياك؟ درناه بعشوائيه بعض المرات لاننا بقينا كندخلوا الابقار وكنستوردوا ابقار ونسينا الابقار ديالنا اللي هي عندها القدره تحمل الجفاف، عندها قدره ديال التحمل بعض الامراض. هادشي نسيناه في الاول. هادو اخطاء درناهم زعما كان مغفورين لينا ولا مغفورين الاساتذه اللي دارهم قبل منا. يعني ما خصناش ننساو ان اليوم الا كانوا المغاربه عندهم الحق انهم يشربوا وهذا هو اللي بغينا نوصلوا ليه في الجمال في النوق وفي الجمال. ما كرهناش ان اي مغربي من جنوب المغرب حتى لشمال المغرب يبغي الحليب ديال الناقه يخرج السوبر مارشي اللي حداه ويلقاه. وهذا هو وهاد التقنيات هادو هما احسن التقنيات. علاش؟ انا نعطيك انا سبيل، لان انا قلت لك تجربه كبيره في الاغنام. هي جرعه واحده من الزريعه ديال الاغنام تقدر تلقح بها 20 نعجه، وهذيك 20 هذاك الجينيتور هذاك الفحل كيكون عنده خاصيات هامه. ما غناخذوش اي واحد لقطناه من شي ضيعه وجينا بدينا كان نلقحوا به النوق. الا خدينا غادي ناخذوا واحد من اللي عنده خاصيات ممتازه، عنده انتاجيه حليب، لان الانتاجيه ديال الحليب راه ما جايش غير من الناقه، راه جايه حتى من عند الجمل، ياك؟ اتفقنا عليها. اذا ملي غادي ناخذوا واحد الجمل جيد بطبيعه الحال راه حنا وكنستعملوا هاد التقنيات. هما احسن التقنيات وهما التقنيات اللي غادي يديونا بعيد. ولكن هذا ما كيمنعش انني دائما كنوصي عليه ان راه هذاك الضيعه الصغيره، هذاك الكساب الصغير، راه هو غادي يبقى حدانا دائما، وتا هو غادي نردوا ليه البال. وهذا الشيء هو اللي طرا في الاغنام. قلت لكم ملي شفتوا السبعينات في المغرب عاودتوا رجعتوا للتاريخ ديال المغرب، غادي تدخلوا لاي ضيعه غتلقاوها مرونه، غتلقاو الكحل الاحمر الزرق، غتلقاو جميع الالوان. ولكن اليوم كتمشي لمهد السلالات كتلقى مهد السلالات محدد، وغتدخل الناحيه ديال برشيد غتلقى السردي، ما تلقاش شي نوع واحد اخر. غادي تمشي للشرق غادي تلقى نوع معين. هاد الاجتماع اللي اليوم حنا اجتمعنا هنا باش نمشيو بهذا القطاع هذا ان شاء الله للقدام، ونوصلوا لهذا المبتغى، نوصلوا لاي مغربي يحتاج لتر ديال الحليب ديال الناقه في الرباط، في اكادير، في طنجه، في العيون، يلقاه حداه. هذا هو المقصود.
أعتقد تكملة الفكرة حول التلقيح الاصطناعي. التلقيح الاصطناعي هي تقنية، ولكن كما جاء في المداخلات يوم أمس واليوم تكلمنا عن القطيع. راه إلا ما درناش تأطير القطيع ويكون القطيع معروف، ما يمكنش ندوزو لهذه المرحلة هذه. اللي نطلبوا دابا حاليا هو يكون مركز متخصص في التقنيات الحديثة للتناسل. وأعتقد هذا نداء للجامعة محمد السادس وللأساري باش يديروا، باش الكساب يجي يشوف بعدا، خصو يجي يشوف. راه في الإمارات ماشي الكساب اللي عنده المراكز، كاين المراكز ولكن الكساب كيجي يشوف بغا ولا ما بغاش. الأستاذة قالت لك في التلقيح الاصطناعي لا بالسائل المنوي المجمد ولا الطازج، الناس ما كيعجبهم هذا، كيعجبهم التناسل الطبيعي. التلقيح الاصطناعي فاش مهم؟ هو في تحسين وتطوير السلاله. هما في الامارات كانوا المره الاولى بداوا التلقيح الاصطناعي على حسب السباق. انت شتي الارقام الخياليه ديال الاثمنه اللي عرض واني خيالي. هذه نتيجه التلقيح الاصطناعي ونتيجه نقل الاجنه. هو نقل الاجنه سبق التلقيح الاصطناعي في المغرب عند الابقار التلقيح الاصطناعي سابق نقل الاجنه. نقل الاجنه في الابقار تقريبا غير موجود في المغرب لان الكلفه ديالو كبيره، والتلقيح الاصطناعي اعطى دفعه كبيره للكساب وللتحسين. دابا حنا خصنا نبداو من الاول. هو نعرفوا القطيع شكون هو، تعلم الكساب يكتب تاريخ ديال القطيع، فوقاش جات، فوقاش مشات، فوقاش مرضت، شكون اللي داوى، وشكون اللي، باش يكون عندنا معلومات. كما قالت الاستاذه بشرى، كتدخل القطيع ما كاين حتى شي رقم، كل شيء في هذا يمكن سيمانه، هذه عامين، هذه ما كاين حتى شي حاجه مضبوطه. خاص يكون نضبطوا الامور ونمشيو بتريث. راه باش دير مركز ديال البيوتكنولوجيا راه مكلف جدا، فهمتي؟ مكلف. لذا خصنا الانسان يتريث. هاد المداخلات العلميه انتم شفتوا دابا الاستاذه كيفاش جات طرحت هذا المشكل ديال التلقيح الاصطناعي. انا راه كنتصنت لها هذه اللولوس كيدمور، كنعرفها من مده زمان، ودائما كاين مشاكل، دائما كتحاول راه ما كاينش وصفه اللي يمكن نطبقوا على التلقيح الاصطناعي. انت قالت لك دابا في الاخر باش جاوبت، سولتها واش التلقيح الاصطناعي كاين في الضيعات؟ قالت لك لا ما يمكنش. واش درتي التلقيح الاصطناعي؟ قالت لك درت كانوا بعض النتائج. واش درتي التلقيح الاصطناعي بالسائل المنوي المجمد؟ دارت واحد المحاوله، فهمتي؟ راه شويه اعتقد الا هضرتي على نقل الاجنه يمكن غدا نبداو، ماشي مشكل. شكرا.
So now we reached the end of this session. I would like to thank heartfully all our speakers for their insightful presentations. Please applaud them again. So now normally we have 1 hour session of poster session, but we have less than that. We have just 30 minutes. So please, the people that have posters, stay near I ask you kindly to stay near your posters for the people that would like to know more. After that, we will have a lunch time and we come back here at 2. So thank you very much to all of you. Yeah, stay. Yeah, just it's not me. So I will let them. Now just when you show the people, you tell them you go forward with the green, backward with the red, and then you want the laser, it's this one.
Bir gün şu dünyadan götürürlerse Bir gün şu dünyadan götür gidersen boşa gider de gözyaşım.